I like experiments you could do with a Petri plate and a toothpick. Those are the kind of experiments I like. And I ended my commentary by saying, my life's quest is fulfilled. I now, after 50 years, I finally know how what I knew 50 years ago actually works out mechanistically. And that's about as cool as it gets.
In today's episode, we speak with Dr. Michael Manson, who's dedicated almost 50 years of his career to understanding the bacterial flagellar motor. In the interview, Manson walks us through the history and key people and experiments that have revealed how biology could create a real working motor. We had such a great discussion that while he was still in town, I actually did a follow-up interview with a 3D printed model of this motor so that he could physically take us through every single component and describe its function in greater detail. And so you can find the timestamp to that follow-up interview on screen now. We'll also be doing a giveaway with several pieces of the model. To enter, all you have to do is subscribe and leave a comment below talking about your favorite thing that you learned from this video. We'll be going through in a few days and selecting our top 5 favorite comments as our winners. Now, without further ado, please welcome Dr. Michael Manson.
The 632 nanometer podcast doesn't have any sponsorships. It's an art project, and we're going to keep it that way. But we wanted to do an unpaid advertisement for Tesla Autopilot. Both of us use it. We've both done probably over 1,000 miles on Autopilot now, and it's just an incredible technology. It's gotten amazing over the last 2 years, and I'm personally very passionate about it because I was hit by somebody who ran a red light, and my pregnant wife was in the car, and it's just something that really shouldn't happen anymore. Like, we have the technology to stop this. If that person was driving a Tesla, it wouldn't have happened. So luckily it all turned out fine.
Driving in Boston is a hell, and I think that definitely helps with finding the right way. It's mind-bending and life-saving technology. I highly recommend.
You have your hands free, your mind free, you can think. Yes, you have to pay attention to the road to an extent, but it really drives itself. But they really improve it all the time. I don't think I've had to take over probably in the last 6 months or something like that. And there's literally all of my driving is on autopilot. Anyway, go out and try this. I think every car should have it, not just Tesla. This software should be in every car on the road.
If you are a fan of the 632 nanometer podcast, you most likely love quantum computers. And now I'm in the lab at CheY Computing, where we build the most advanced neutral atom quantum computers. We have plenty of job openings, and I'm hiring for the position of the quantum machine builder. If you have a relevant skill set and want to contribute to the race for building the first fault-tolerant quantum computer, join us and enjoy the rest of the episode.
I've been working with bacteria. Bacteria actually have a behavior, and that behavior is chemotaxis. Bacteria swim, and they swim in a purposeful way. They swim to get to places they want to be and to get away from places they don't want to be. And Howard Berg at that time had just published a paper showing that the bacterium E. coli swims in a random walk. It swims in a straight line for a while, and then it tumbles around and goes off in a new random direction. And so it does a 3-dimensional random walk, just essentially accelerated diffusion. But the reason that's useful is that if the bacterium is swimming in a direction it likes, it swims for a longer time before it tumbles. And then when it tumbles, the new direction is random. And if it's not good, then it tumbles sooner. And if it starts going the right way again, it gets longer. So what happens is you get a biased random walk that gets you to where you want to be, right? So that was the paper that...
Just curious, what is I'm more from solid state physics, there is the electrons when they bounce, there's a mean-free path. So for bacteria, what's the distance, the average distance they usually travel?
Well, they typically will run for a couple of seconds, and they swim at 10 body lengths a second. So I would say 20 to 50 microns probably. So, 20 to 50 times their body length before they tumble. Mm-hmm. And that's their behavior in the absence of any stimulus, right? If they see things they don't like, they tumble more often. And they get away from them by tumbling more often. And when they go in the direction away from it, they go longer, right? So they can go towards things or away from things using this biased random walk. And actually, Howard Berg, it turns out, wrote a book called random walks in biology, which is a classic of the role of diffusion in biology, right? So the bacteria are not just diffusing. If they diffuse, they just jiggle around, right? But because they're too big. But essentially, they're doing accelerated or powered diffusion by doing this random walk of running and tumbling. So that was the thing, and that's what he talked about. And I thought, well, this is pretty interesting. And because my PhD advisor was his host, there was a party at his house that night where I got a chance to talk to Howard Berg. And I thought, wow, this would be so cool. Bacteria have a behavior, right? You could study. And by that time, I'd learned if you want to study behavior in birds, you have experiments that go over months and years and are very difficult to quantify accurately. But I know with bacteria, because of what I've done, you could do experiment overnight. You can grow up the bacteria overnight, right? And you can design experiments one day and carry them out the next week. So I said, okay, I can do that with bacterial chemotaxis. And talking to Howard Berg that night, I decided, okay, that's where I'm going to go do a postdoc. Okay, that's what I want to do, because I can take, and he knew nothing about bacterial genetics or any of that stuff, which I did know. I knew nothing about biophysics. So it was, you could say a marriage made in heaven of two different expertises coming together.
So you did postdoc in his group, but working on bacteria while the main group was focused on behavior of those?
I met him in 72.
I see.
In 73, it got even more exciting because in 1973, he published the paper in Nature that bacteria swim by rotating helical filaments. So everybody had thought that rotation was impossible in biological systems. But being a biophysicist, he knew, no, look, you can't rotate your arm. You can't rotate around a joint. You can't rotate around blood vessels, nerves, whatever. But if you're molecular, at the molecular level, molecules can rotate just fine. And they don't need lubrication either. They just can rotate. And so he proposed that, the only experiment he did to show that was he got two cells stuck together by their flagella, and they rotated in opposite directions from each other. Okay? That is only evidence that the rotation.
Because when you look at it on a microscope, like back then, you would just see the flagellum kind of moving, but you don't know that it's rotating around an axis.
If you look at the flagellum, if you could get high enough resolution to see it, and you could with high intensity dark field microscopy, you could actually see a helical motion, but you don't know if that's a helical beat, or a rotation of a helix, right? They look the same. When you say
beat, you mean like something like this, or just up and down?
Well, if you take a rope and spin it like this, right? You get
I see.
a helix pattern. And looking in the microscope, that looks exactly the same as if this were rotating, right?
Yeah, actually rotating on an axis. Yeah.
And he had all sorts of clever toys that he used to illustrate this in a lecture.
So wait a minute, if you have two bacteria and the flagella are intertwined like this.
Okay, stuck together, yeah.
If they're both beating, you're saying they won't spin off in different directions.
No, they wouldn't spin, they just thrash around.
Yeah, but if they're spinning,
rotating, they're going to spin
like two screws almost, or whatever.
Yeah.
That's really interesting.
I mean, so
how did you hook them together?
Randomly.
They just stuck together randomly.
Just like
he might have used antibodies. I mean, you can
really
bivalent antibodies. An antibody is like a Y-shaped molecule, and the two things bind equivalent stuff. So if one binds to one flagellum, the other binds to the other flagellum, you'll stick them together with antibodies, and they'll be stuck together, and then you can see the spinning. That's how you set them down on a surface. When we look at rotating cells, you use antibodies we used to use antibodies. Now they've developed sticky flagella that stick to glass.
So you're saying you have an antibody and that holds the flagellum, and then the cell rotates around it.
Yes.
Oh, wow, interesting.
And that came soon after. He didn't know that at the time of his first publication, but within a year or more, several people had started looking at this.
Wow.
They call them tethered cells, right? You stick them down by one flagellum, and they spin And it turns out that's absolutely critical for what Arv and Samuel did in 2025, because they were looking at tethered cells. Because you have to have them in one place in order to do the kind of microscopy they did. You have to be able to focus on one place. So they looked at a motor of a tethered cell, right? That's the way they could get the resolution that they needed to do what they did. So anyway, that was, so now you have, and then what you learned, the next thing that also came out of Howard Berg's lab, and this is all before I even got there. This was all happening after I decided to go there, but I was taking a long time to finish up my PhD because I had a biking accident and broke both my wrists. So I had 2 casts for 6 months, which didn't make it very possible to do experiments. So it took a relatively long time to finish my PhD.
Were you managing like other younger grad students?
No, I was basically writing and doing nothing. So. And then I would be playing volleyball, which sure didn't help. I could really smash the ball, but it apparently didn't help the wrist heal. Anyway, I was known as the phantom postdoc in the Berg lab because I was supposed to get there in 1974, and then went to 1975, and I didn't get to Boulder, Colorado, which was where Howard Berg was at the time, until I think November of 1975. So like a year and a half after I wrote an NSF postdoctoral fellowship. And interestingly, what I wrote it for had nothing to do with rotating flagella. The idea was, how does how the cells sense their chemical environment, okay? How does that signal get from the receptors that sense a chemical to the motor, right? And at that time, it was an attractive idea that the signal might be electrical. And a guy named Dan Koshland, who was a very famous professor of biochemistry at UC Berkeley, and was sort of a competitor of Howard Berg's, liked the idea that bacteria were like little nerve cells, right? So there'd be electrical conduction. And actually, I wrote my postdoctoral fellowship to test that idea. And I spent a lot of fruitless time trying to look for changes in membrane potential with potential sensitive dyes in an optical device, and I wouldn't see anything. What I'd see is, though, when I added whatever I was adding that attracted or repelled, there was a big change in the membrane potential of the cells, but it turned out it had nothing to do with the chemical. It was just because I was putting in oxygen, and I was increasing the proton motive force because I was putting in oxygen. And that's what finally got me, after wasting, not wasting. When you show something is incorrect, I guess you're not wasting time. But that is what got me to think of the idea, well, maybe we should look at the idea that the membrane energetics is what's driving the flagellar rotation. And that's so I started doing those experiments.
And so when you got to the Berg lab, was it already known that there were processes being driven by protons transiting the membrane of bacteria?
Yeah. So
like when did
Peter
that
Mitchell,
fundamental idea?
Peter Mitchell, who was thought to be crazy. He was kind of a hermit who did his research in a lab in a castle in Cornwall in England. Okay. He was the one that proposed that a proton motive force, electrochemical gradient of protons across the membrane, drove all kinds of things. And people, I remember from when I was in grad school, saying, how does electron transport work? How does transferring electrons along the membrane convert into making ATP? That was a big thing, the coupling between electron transport and ATP synthesis. And what he proposed was, well, what the electric transport chain does is build up a proton motive force. And then that proton motive force is used to make the ATP. Okay? So it's a two component.
How did he have that insight? Like, I guess at the time it would have been
He's a
very
genius. And everybody thought he was crazy. I mean, they thought he was crazy. And he was not particularly articulate. And somebody that I got to know who was at the Jewish hospital in Denver when I was in Boulder working with Howard Berg, named Frank Harold, actually, and he would say until the day of his death, the most important thing he did was make Peter Mitchell understandable to people. Because he wrote a review article explaining what Peter Mitchell had done in a way that somebody could understand, right? And he no doubt was instrumental in getting Peter Mitchell the Nobel Prize. And because of that, there's the interesting story, how things work in biology. You know, it's all the connectivity, right? I happened as an undergrad to walk into Hilliard Berger's lab at Johns Hopkins because he had to work in a lab as an undergraduate. I went to Johns Hopkins because I had the Chesapeake Bay Institute. I thought I could study marine biology. I walked into this lab doing molecular biology and E. coli. From that, blah, 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 Hilliard Berger had been a postdoc with Charles Yanofsky, blah, blah, blah. Howard Berg came and gave a talk at Stanford, and I met him in Charles Yanofsky's living room. And that's how my scientific career developed, right? Just happenstance jumps from one thing to another. So it's, you know, as I think Jim Watson said, you don't have to know physics. You just have to know a physicist, which was, in his case, Francis Crick, right? Because Jim Watson didn't know physics, right? But collaborating with Francis Crick, they worked out the structure of DNA, right? So these things are important. And it was same with Howard Berg. I was scared shitless when I went to Boulder because I didn't know biophysics at all. You know, Jesus, you know. And I thought that this is going to be horrible. But he didn't know anything about bacterial genetics. So we were equally ignorant of what each other did or knew. And it turned out to be perfect, right? Perfect complementation. But the way that worked was through Frank Harold. Frank Harold, it turns out, worked with a bacterium, Staphylococcus, that doesn't have an electron transport chain. Okay, there are bacteria that don't use the membrane ATPase to make ATP. They make ATP through substrate level phosphorylations as they break down sugars or whatever they're metabolizing. And the nice thing about that is that they're really easy to starve. E. coli builds up all kinds of storage products of carbohydrates that it can use. So if you stop feeding it, it'll swim for hours. But this Streptococcus will stop doing whatever it's doing. And I just happened to read, going through Journal of Bacteriology, that there was a motile Streptococcus that had been found in cow dung in the Netherlands.
So the experiments were mostly on E. coli and Streptococcus. Any other bacteria contributed to like some insights?
Those two I've worked with are E. coli and Streptococcus. Lots of other people work with Salmonella, which is closely related to E. coli. Some people have worked with Bacillus subtilis, which is a gram-positive bacterium that is motile, but also has an electron transport chain. But the nice thing about the motile Streptococcus was that it didn't have an electron transport chain. So you could starve it, and it stopped swimming within minutes. Okay, just took away a carbon source, and it stopped swimming within minutes. The other great thing about it was a gram-positive bacterium. So gram-positive bacteria do not have an outer membrane. The outer membrane is a pain in the ass if you're using membrane-active agents because they get embedded in the outer membrane. They never get to the cell membrane. But the Streptococcus didn't have that outer membrane. So you could add ionophores, things that let ions through a membrane, to them, and it would get directly to the cell membrane. And that was the idea that I had, right? Inspired in part by Frank Harold, who worked with Streptococcus. So I said, look, we should be able to do this. We should be able to starve a Streptococcus and then create a potassium diffusion potential in it by just adding a potassium ionophore, something that conducts potassium across the membrane just in response to the gradient of potassium across the membrane. And if you have a cell that's loaded with potassium and you add valinomycin, what's going to happen? Well, potassium is going to leak out, right? Down its concentration gradient. What does that do? Potassium is electrical charge. It's going to set up a membrane potential, interior negative. What's the membrane potential going to do? It's going to attract positive ions to go into the cell, right? Mm-hmm. And so that's the experiment we did.
And so then protons start flowing again.
And proteins start flowing in, and then these starved cells that are just sitting there start swimming. Okay, that was the experiment that we did. It was published in 1977 in Proceedings of the National Academy.
How did that prove that protons were the source? Because it could have been some other positive ion.
It could have been some other ion, yeah. But you could do it. It turns out you could do it also. The proton motive force works not just with electrical charge, it also works with pH, because difference in hydrogen ion concentration will drive it. So you could show if you just lowered the pH outside, that you could get them to start swimming too, because that would drive hydrogen ions in as well. So we showed that you do either electrical potential or a chemical potential of hydrogen ions, a pH gradient.
So that kind of triangulates it.
Yeah.
So you said you need to kind of inject this potassium into the...
No.
Sorry, not potassium, but the...
The linomycin? No, it's just, you just add it.
Inside the bacteria?
It just, not in, no, you just add it to the medium and it gets absorbed to the membrane.
Oh, I see.
It just, it's an ionophore. Ionophores are things that conduct ions across membranes, so they have to have the ability to insert into a membrane, but they also have to have a channel in them that allows a hydrophilic channel that allows the ions to go through.
Is it a protein or what is it?
No, it's a small molecule.
Oh, okay, but it basically sits in the membrane. It opens up a little pore for potassium to pass.
And potassium will flow through either way. And that was the thing. Okay, now to decide, about the energetics of this. So this was the paper we published in 1980. We found it, fortunately, the Streptococcus, if you sheared off their long flagella and just left little stubs, and the same is true of E. coli, that they'll sit down on a surface and get stuck by their flagellum, and then the cell body rotates around one place, right? They go one way, and then they go the other way.
But when you say shear, did you shear it off just by putting them in a blender?
Yeah,
or
exactly, in a blender. Yes, exactly, in a blender. And you have to be very careful if you want to look at actual swimming cells. When you get them out of their growth medium, you have to be very careful how you centrifuge them and resuspend them so as not to break off their flagella. Anyway, they'll just stick that. And fortunately, we didn't have an antibody against the flagella for the Streptococcus, but they just stuck to glass spontaneously. And then the cell bodies would turn. And they would turn one way, and then they'd reverse, and they turn the other way, and they turn one way. And just like with E. coli, one direction of spinning was smooth swimming, and the other direction of spinning was tumbling. And the reason that happens is because the flagella are left-handed helices, and if you turn them counterclockwise, they form a nice bundle. If you start turning them clockwise, that bundle flies apart, all the flagella will go off, and it just flails around, right?
So meaning like a bundle of flagella, which if you're turning them one way, they all kind of spin together.
Yeah.
If you separate them, then
flies apart.
They're spinning, they're pushing in different directions.
Yeah. And that's what causes the tumble, causes the reorientation.
Is there any choice of where it goes, or it's random?
No, it's just random. It's just in some new random direction. They start turning the flagella counterclockwise again, the bundle reforms, and they swim off in that new direction.
So it's only like stop and go. That's the only...
Stop, go.
Random, okay.
Go, yeah. And Howard Berg developed a microscope that showed that. He developed a microscope that could keep a bacterium in a microscope in focus, right, by moving the stage to keep it in focus in the microscope. And if you record the motions of the stage, right, that tells you the path of the bacterium. And that's how the automatic tracking microscope, that's how he showed that they ran and tumbled. I mean, Howard Berg was an instrumental genius, an absolute instrumental genius. At Colorado, there was an old German physicist, old German machinist that ran the machine shop. And Howard Berg was the only guy that got to use all of the sophisticated equipment. Nobody else, students or faculty, they had a separate little place where they could, have simple equipment. But Howard got to come in and use the most sophisticated equipment because he was that good at it, right? And even the very particular German machinists had to let Howard Berg use. That's what Howard would do. That's his favorite thing, was go to a machine shop and start building something. That's what he really loved to do, and he was really good at it. So these guys are tethered, right? But so with E. coli, it was shown first with E. coli, but it was true for for the Streptococcus as well. They turn one direction, and that's so. If you're looking at it, if the flagellum would be turning counterclockwise, the cell is going clockwise, right? If you hold the flagellum still, and then when they reverse, when the flagellum would be going clockwise, then the cell goes counterclockwise. And so you could show that, and you could show that if you added an attractant chemical, that they spent a longer time going in that direction, right? And you could very easily quantify by looking at the time they did that until they adapted to the signal. And with the Streptococcus, they would do the same thing: turn one way, turn the other way, turn one way, turn the other way. And so what I did was I set up different magnitudes of proton motive force. Had starved cells, they were just sitting there tethered, they just wiggle around. And then you would add dolinomycin with different external potassium concentrations, right? So you get a different magnitude of the potassium diffusion potential, or with different pHs. And they'd start spinning, okay? And you record the speed at which they spin. And I heard this blob of data. It was very hard to see anything. And Howard I said, get off the pot, plot your data, see what's really going on here. And it turned out when you did that, even though the individual data just were looking like a scatter plot, that you got very clear trends at the different proton motive forces. And if you then corrected for the length of the cell, because what caused all the scatter was that small cells turn much faster than large cells, right? Because it turns out that the Flagellum motor operates at constant torque, which means it's going to turn a long cell much slower than it turns a small cell. So all my scatter was due to that. And when I corrected for the length of the cells, they formed really nice plots, right? Linear plots with the proton motive force and the speed. And that was our paper that we published in the Journal of Molecular Biology, showing that, in fact, it's called the energetics of flagellar rotation, that there was the speed of rotation was directly proportional, at least at this range, this high load. This motor is really working hard, right?
It's a single motor spinning the whole
cell. Yeah, but it's spinning a little flagella, right? But, and they go like 300 rotations per second. These guys are turning at, one to a few rotations per second. So they're working at really not where they would normally be working in their normal function, but in that range, they're operating at constant torque.
Yes, next question is what switches the direction of the torque?
See, now you're getting to the crux of things. Okay, that's one crux.
I see.
That's the crux from the paper of 2026, right? So Arvind Samuel published these two papers. His lab published these two papers. One was the one in December of 2025, where he showed that these little stator units, which we'll talk about in a moment, rotate. And then in March of 2026, he showed that a single molecule of the signaling molecule, CheY phosphate, a single one of those binding to the flagellar motor causes it to reverse direction. Okay, now that will require another whole set of things to set up how that works. But maybe to stick with the motor, okay, first. And as I say, to get into the details of, okay, I have to back up a little bit. So what we knew, and what was known for a long time, was that you could make a complete flagellum. And if you look at your picture, right? If you look at this, you can make all of this, right? All of that structure. If you don't have these little guys, it doesn't turn. It doesn't work, right? So you need these two proteins.
How was that known? I mean, people were purifying.
Oh, you could show, you knock out the gene with mutations, and then you'd express the protein from a plasmid.
At what point were all the components known? Like even
quite early.
How did that get put together?
Oh, there's a group in Japan that put that all through. They did. I mean, as you can imagine, there are like, I don't know, 30 different proteins that go into making this structure. It's a very complex structure. And it has some really incredible aspects to it. Like you have to export everything that gets built on to make this flagellum, has to go out through the middle of it, diffuse out through the thing. But first here you have this thing called the flexible hook. What does that do? Well, that allows rotation that's perpendicular to the membrane to bend around. It acts like a universal joint, right? So it allows rotation here to turn into rotation there. And that's of a fixed length. All of this down in here, inside the cell, all this that goes through the cell membrane, out through the outer membrane in E. coli, all of that, including, and this yellow part out here is the flagella, which is what you see here, how long that is, right, relative to the cell. This hook is just a little short thing, but it is of a fixed length. And at some point, the machinery at the base here knows. It switches from exporting the proteins that make up this hook to exporting the proteins that make up the flagellum. And actually, there's a protein called the ruler protein that measures that length. And when it gets to that length, it essentially tugs on something down here that changes the specificity of what this export apparatus will export.
But so when you say export, what's really happening, it's not growing from the base. It's actually...
No, it grows at the tip. Everything grows at the tip.
It's exporting these proteins. They kind of diffuse their way to the tip.
They diffuse through the center. The center is hollow.
Oh, the center is hollow. Okay.
And these guys assemble at the distal tip.
Okay.
Right? And there's a cap protein in each case for the hood and for the flagellum that keeps them from just going out into the medium, right? It captures them there so they get assembled there.
But you're saying this was known in the 80s?
Oh, this was known before that.
But how could they have known all these structures? Like, how did they come up with all this like mechanistic?
Genetically, right?
Oh, by killing different parts.
Yeah, you just knock out different genes, and each gene, when you knock it out, the structure will get made to the point where that protein is needed, and you'll see that intermediate structure. So you can go through them, knock out the genes one by one, and see how the pieces.
And is this with like electron microscopy? They were looking at the structures, or
just simple, electron microscopy.
Just to give like maybe a taste of the complexity of the problem, like for the gene code of the bacteria, like how long is it, and like how many permutations you need to do there?
E. coli has about 4,000 genes. It has the circular chromosome of E. coli is about 4.5 million base pairs, and that encodes about 4,000 genes. So roughly one gene per 100,000. Per 1,000 bases. By the way, that's way denser than in humans. Like 1% of your genome encodes proteins. In E. coli, it's like 90 or 80% of the genome encodes proteins. Right? We have big spaces between our genes. We also have introns in our genes that have to be cut out before the proteins get made. Anyway, they did that just laboriously genetically, knocking out these genes one by one and seeing what structure got made. It was a lot of work.
Knocking genes, what is it, like chemically? What do you do?
Mutation. You make a mutation. Oh. You could now you can do it directed, but back then they weren't doing it directed. They were just randomly mutagenizing and get something that didn't swim, and then look at it, and then map the gene onto the chromosome, right? And that way they worked out how many genes there were, how they were organized in the chromosome, which genes were next to which ones.
Man, sounds like a lot of work.
It's an enormous amount of work. A guy named Iino in Japan was the leader in doing this and established all this. It was amazing work. So you've got convergence of a lot of different things here coming, right? Mm-hmm. Different people doing different things.
Without this information, it would have been hard to guess what's going on.
Right, you could have done it. Yeah. But it turns out, just the paper. I just reviewed the paper that says how this switch works. That when this gets to the right length, there is one protein down here in the export apparatus that changes conformation. and it goes from having, it's in a non-numeric ring. It goes from one conformation of that ring that allows things for the hook to get out to another conformation that allows things for the filament to get out. And that is triggered by this ruler protein that when it gets to the right length, it triggers the change in the export apparatus that switches its export specificity. That paper, I don't think has been accepted yet, but hopefully it will be.
Incredible.
Just out of curiosity, was this mechanism adopted, I don't know, somewhere in vehicles or other motors?
Well, no, because the thing is, remember, the thing here is that you don't need any lubrication, right?
That's right.
At the molecular level, all this rotating stuff, you don't have to worry about things grinding against each other or anything like that. So, to adapt this to any kind of motor on a scale that would be useful for human things, there'd be a lot of problems that would have to be solved, right? Because you could do things at the molecular level that are very different than what you could do at the macro level.
So I guess in the 80s or even earlier, you had the different components, but nobody knew what shape they were. I guess it wasn't known like what was the shape of all these proteins.
Oh, you mean the individual structure of the proteins? Yeah, that came out with time as that was basically mainly X-ray crystallography, right? Mm-hmm. You have to crystallize these proteins and determine their structure, their X-ray structure. Now you can do really high-resolution EM to get structures of proteins. But it used to be you had to be able to crystallize a protein. Not every protein crystallizes, so that was a very tedious, long process as well. But all this got worked out, the structure of all of this got worked out by making mutations and looking at where the structure stopped developing. Mm-hmm. Fortunately, the thing goes at a sequential basis, right? So, if you have a mutation here, the base down here is called the rod, which rotates, which is connected to this, which rotates. Well, you have like 5 different proteins there, and you knock out different ones of them, and you have a rod of a different length, and so forth. And so it's just, it's amazing, the amount of work. Okay, but here's the critical thing. So all of this, as I was saying, all this down here, all of this gets made. But if you don't make these 2 proteins, which are called Mot proteins for motility, if you don't make the Mot proteins, the motor, you make the flagella, but it doesn't spin. Flagellum looks perfectly normal, but it doesn't spin. But if you take a mutant that has knocked out these proteins, and you now express those proteins from a plasmid where you've cloned the gene for it, suddenly the motor starts rotating. And we did a lot of work on study because it turns out that this this part that in orange here is connected to the cell wall. So it actually anchors to the cell wall, which is a firm structure. So this is firm, and this part has to move. The part down here has to move, right? And we didn't know how that happened. We knew that the orange guy had to attach to the cell wall. We did some work on that. And we knew it had to be in the right orientation because we had mutations, point mutations, ones that just change a single amino acid that would make it non-modal. And then we could get compensating mutations in structures here that would restore the motility. So you got the idea. It had to be in the right spatial relationship, right? You move it a little bit, it doesn't work. Then you move the other one, and it works again, right?
Were there proteins that were part of this complex that you could break and they would still work?
If you knock out any one of those proteins, the thing doesn't work at some level. I mean, some of them, if you knock them out, you don't make anything. Right? The ones that are involved in the very early stages of assembly.
But you could know the syndrome. So if you delete like this part, it's not doing one thing. If you remove the other thing,
a whole bunch of them.
Like
If you knock out the gene, you don't get anything, right? Because there are certain things you need just to get started in making it, right? And though that's not very informational because they just know, they don't let you even get started. But all these other steps, through making the rod and all the casing around it, the L&P rings that are around here that form a bushing to allow the rod to go out through the cell wall, and all of those.
I mean, one thing that really surprised me about this is that you can have this spinning axis, but it doesn't leak protons through that hole.
No, it's got to be tight, right? It's got to be tight.
It's got to be extremely tight.
You cannot have a hole. A hole in the membrane knocks out the proton motor force right away, because protons just flow through a hole. And we'll get to that a little bit about some work we did with this motor protein. So we knew the motor protein was necessary. We knew you could add it back after the flagellum was complete. And it turns out later work showed that most of these guys are just floating around in the membrane. So it's a membrane protein embedded in the inner membrane. It doesn't attach to the cell wall when it's just floating around in the membrane. Something about it's engaging with the motor causes this orange part to stick up and attach to the cell wall, and then it stays in place, because it's called the stator of the motor, because it can't move. It's attached to the cell wall, right? So, and you need that, right? You have to have something attached to the cell wall in order to have something to push against for the flagellum to turn. And it's this protein that does that. And it turns out even one of them is enough. And when you're turning a free-floating flagellum, you probably only have a couple of these on the flagellum. When you're turning it against a really high load, like with a tethered cell, then you have up to 10 or 11 of them.
Yeah, I thought when reading, in preparation for this, I thought it was amazing that the cell can respond to viscosity and its own size.
Yes.
To increase the number of stators around the
greater the resistance of turning it, the greater the resistance of turning, the more of these units.
How does that control system work? Like, how does it know what the resistance is, or like what is that well understood?
It's not really well understood. There's a lot of work done on it. It's work that my colleague Pushkar Leila at Texas A&M in the chemical engineering department has worked on. Initially, when he was in Howard Berg's lab as a postdoc, and then after he came to Texas A&M, it's something about the torque, the twist on the protein that causes this part that binds the cell wall to be released. We spent so much time trying to see this protein if it would attach to the cell wall. We could get other proteins we knew attached to the cell wall. We could show that they were attached to the cell wall. We would purify the cell walls. You could purify called the murine sacculus, just the cell wall. You could dissolve all the rest of the cell, and that stays intact. And proteins that are stuck to it are attached to it. But this protein, we never found it. And it turns out the reason is because they're never attached to that until they're around the flagellum. They can be floating around the membrane as much as you like. Okay? Then one other thing we did was we showed that if you made a truncated version of this protein, this orange protein, okay, that didn't have the part that bound to the cell wall, and you expressed that in the cell, that the cells stopped growing. And then we also showed that there was a little stretch of about 10 amino acids right after. So the way this protein is made, there are two central proteins, the MotB protein, the dimer, and as we'll see, there's a pentamer of the MotA protein around that, right? And there's right after the membrane spanning thing. So the N-terminus of the MotB protein is inside. It goes out. Right after it gets out through the membrane, there's a little stretch, which we call the lid. If you just deleted that, those 10 amino acids, or even mutate one of them that's critical, what happens is that when you overexpress that protein, suddenly the cells stop growing. You could overexpress the normal protein as much as you like. Nothing happens. Overexpress the one that lacks that lid or has a malfunctioning lid, and the cells stop growing. And the reason is because now the proton channel is open. What that lid does is close the proton channel. Okay? So when these things are just floating around in the membrane, the channel is closed. The channel only opens up when you attach around the motor, and that pops off the lid. So the only time it's conducting protons is when you're around the motor. So you can make as much of this protein as you want, these proteins as you want. They're floating around the membrane. They don't do anything. But if you make it without the lid and you overexpress them, now everybody's letting stuff through. And we showed that you actually acidify the cytoplasm. The protons that are coming in, and that's why the cells stop growing.
Is it only protons get inside the cell or some other ions as well?
Through this, only protons. Now, when we say protons going through, I should have a caveat here, which is that it maybe what's actually going through are hydronium ions.
Right, H3O.
H3O. Because there are motors that work with sodium, and there are motors that work with protons. Turns out the sodium ion is just about the same size as hydronium ion. So actually, it's easier to imagine for me that it's actually hydronium ion that comes there and binds to that critical aspartic acid residue. If a sodium can do it, then why not a hydronium? And it turns out that there are a few rare ones that can use potassium to drive the flagellum. So the critical thing is neutralizing the charge of one aspartic acid residue that's in the middle of the membrane-spanning region of that central MotB protein. That neutralization is required for the motion.
And it sounds like, because there's always a membrane gradient for protons, it sounds like this is always on. Like once you have this complex, it's always going to spin.
Oh yeah.
And is there something that regulates that, or it's just generally on?
No, it's always on.
It's always on. So that's what.
Well, there's some other work now that shows that there are other things you can use to stop it. And in some organisms, that's quite well studied. Like, for example, in Bacillus subtilis, which swims around when it forms a biofilm, there's actually a protein that binds the motor and stops it from turning. Because it turns out if you want to make a biofilm, you don't want to be swimming around. Right. So
there's
you can actually shut it off.
Additional
mechanisms that can shut it off. Right. So yeah, this is a pretty sophisticated machine.
Maybe one more question. So if the proton channel is opened, why the cell stops growing? Is it messes up the
No, it just lets
the membrane potential?
It lets so many protons or hydrogen ions through that it acidifies the cytoplasm. You just overwhelm the ability to pump them out fast enough. So you lower the pH, and if you lower the pH, the cells stop growing.
Well, and just on a basic level, isn't ATP synthase runs on the same protons?
Oh, yeah.
Right? So basically the core thing that's like powering the cell also runs on protons. So if you're short-circuiting you're basically short-circuiting the whole energy system.
You are.
If you make a hole in the membrane.
No, you mentioned the ATPase. That it was shown that rotated much later than the flagellum. And the way they did that, so this is so cool how this was figured out, that once the structure of the ATPase became clear that there was a proton channel. But the proton channel in the ATPase is a half channel open, leads to an aspartic acid residue in a ring of 10 to 12 proteins. That ring cannot go into the membrane if that aspartic acid residue is charged negative. If the proton comes in, binds to it, now it's neutral, and it can go into the membrane. So what happens is it comes in here, all of the guys are protonated, and over here, it comes off into the cell. So a half channel facing outward and a half channel facing inward. And it turns out that's going to be the same in the flagellum, right? And so it comes in, all these guys are protonated, it turns, and it goes off here. That means you rotate this ring in one direction. And if you think about making ATP, right? ATP, it's three reactions, basically, that the enzyme has to do. It has to bind ADP and inorganic phosphate. It has to combine them to make ATP, and it has to release ATP. And if you think about that 1, 2, 3, that will only work in that in that order if you rotate in one direction. This trimeric ring, it doesn't work in the other direction. If it goes in the other direction, what you do is you split ATP into ADP and inorganic phosphate and release it. And if you do that, the ATPase pumps protons out.
What makes the handedness? Is it the shape of the? Is there like a corkscrew shape in the ATP synthase, or like what
No. It's a trimer.
But why does it go clockwise instead of counterclockwise or whatever? Like what's
Well,
pushing?
because of this ring that I'm talking about here, if protons come in from the outside and bind here, the ring has to go around that way and then before it releases. If protons come from the inside, they bind here, then it goes around that way and releases to the outside.
The ring is kind of screwing into the membrane, basically.
No, it's just in the membrane. It's just
Then
rotating.
Why does the rotation in one direction push the?
Because there's the two half channels, right? If it's rotating one way, the half channel that's open to the outside is accessible, right? And you don't release until you get around to the other half channel. The other way around, the protons come in here because it's just the difference in the energy of the protons. If the inside is negative, the protons are going to want to come this way. If it's not, they're going to come the other way, right? And so this ring that I'm talking about rotates in the two directions. And if they're coming in, it rotates in the way that makes ATP synthesized. And if it turns the other way, if you're binding ATP, breaking up ATP, and releasing ADP and inorganic phosphate, then you're pumping protons out. And it turns out that cells, even like Streptococcus, that don't have an electron transport chain, the way they create their proton motive force is by hydrolyzing ATP and getting pumping protons out through the ATPase. Okay, so it's an ATP synthase or an ATPase. If it's making ATP, it's ATP synthase. If it's hydrolyzing ATP, it's ATPase. And the reaction strongly wants to go in the direction of breaking ATP. That's the energetically favorable way. It's what powers our muscles, for example. All kinds of things in the cell are powered by hydrolysis of ATP and ADP and inorganic phosphate. So you have to have enough of a proton motive force to overcome that energy that it requires to form that bond. If you're below, the proton motive force goes below that, the ATPase doesn't work. Now it'll start hydrolyzing ATP and pumping protons out. And if cells lose their proton motive force, that's exactly what they do. They start hydrolyzing ATP and pumping it out. If cells are growing, for example, anaerobically, E. coli can grow anaerobically. It's not very efficient because it's like the Streptococcus. It can only use substrate level phosphorylation to make ATP. But if it's doing that, then it has to use its ATPase as an ATPase to hydrolyze ATP and pump out to make the proton motive force. Because the proton motive force is needed for all kinds of things besides driving the flagella. It's required for all kinds of transport systems, lots of things. You need a proton motive force, even if you're growing anaerobically where you don't have an electron transport chain. And there the ATPase serves to create the proton motive.
So it's interesting you're saying that this proton motor force is created by external flow of the protons instead of the bacteria using its own energy, like ADP, releasing it and then powering the body.
So you have the way an aerobic bacteria. I mean, your mitochondria are like this, right? Your mitochondria have an ATPase in the membrane, same basic thing that the bacterium has. They're always making ATP, right? Because you always have the proton motive force across the membrane of the mitochondrion, and you're always having protons moving in, and so you're always turning the ATPase in the direction that makes it an ATP synthase, so you're making ATP. So in the membrane of the mitochondria, you have an electron transport chain that pumps protons out. In the membrane of the bacteria, you have an electron transport chain that pumps protons out. So, I mean, the energetics of that is really, really kind of cool.
Yeah, it's awesome. So coming back to, I think we were like in the 80s at this point. So let's continue.
Okay, so the way they showed the ATPase worked is that the ATPase, they took out the membrane part of the ATPase, right? They just took the part that rotates. But there's a rod coming out of it which communicates with the ring that turns in the membrane. And they stuck actually a filament onto that. I forget what it was actin or something. With antibodies, they stuck a filament onto that. And when they added ATP, they could see that filament started turning. So when this ATP started hydrolyzing ATP, it turned this rod. That would be in the direction it would do when it was pumping protons out, right? There's no membrane here, but you just add the ATP and it starts this turning in the direction that would pump protons out. So that showed that the ATPase rotated. And that was, for us, really exciting because we thought, wow, maybe these work the same way. The flagellar motor and the ATPase, it turns out they don't work the same way at all. But they both rotate in response to the flow of protons through the membrane. But the mechanism is totally different.
Any other mechanisms which are similar to this kind of rotation chemistry? Or?
Well, I edited a series of articles for Frontiers in Microbiology. It was called Biological Rotary Nanomotors. They're like 20 articles. And it covers all of the molecular rotary motors. It got experts from all over the world to write chapter about what they study. And it turns out that the structure that's like the motor proteins of the flagellum exists in a lot of other forms. So as we'll get to, I presume in a moment, the high resolution EM, cryo-EM showed that the Mot complex consists of 2 central proteins and then 5 MotA proteins that are around the outside of that, right? That 5+2 structure exists all over the place. Presumably, it rotates because it's all in motors that are driven by the proton motive force. But these motors are coupled to the most diverse things, transporting things through the outer membrane, affecting the way proteins that are involved in cell wall synthesis are made, powering the gliding bacteria that glide along the surface without flagella at all. The same motor, the same 5-2 motor, is found in all of these contexts. And that's one whole set. And then there's the other set, which is the ATPase, which is totally different. Has a different stoichiometry, different chemistry, totally different. But the thing it has in common is that it lets protons through the membrane and causes something to rotate while it's doing that. Now, this comes to the part that's really relevant to directly what we're talking about. But before I do that, maybe I should say a little about the chemotaxis. So there's a cluster of receptors at one or both poles of an E. coli cell. And their E. coli has 5 different receptors. And they're in a mixed patch. Now, what these receptors do is they bind molecules on their outer surface, right? And they send a transmembrane signal that affects the activity of an enzyme on the cytoplasmic side. And what that enzyme does is it phosphorylates a small protein called CheY. Okay? And CheY, in its phosphorylated form, binds the flagellar motor and causes it to go clockwise. Okay? So you have no signal, no receptors, no anything. The motors will always turn counterclockwise. The cells will just swim straight forever. In order to get them to reverse direction, you need this CheY protein in its phosphorylated form. So you also need everything else that's involved in phosphorylating this. If they're missing, then you don't get phosphorylated CheY, and the motor always turns counterclockwise. But somehow when CheY phosphate binds to the motor, it causes the motor to turn clockwise. And we knew that for a long time. We knew that only the CheY protein was necessary to change the direction, because you could overproduce the CheY protein without any of the receptors or any of the other enzymes. You could overproduce the CheY protein under conditions which it would be phosphorylated, and then you would get cells that would turn their flagella clockwise. So you knew that all you needed to turn the flagella clockwise was CheY phosphate, right? But how that works, but it's critical because what the receptors do is they send a signal that affects the activity of this enzyme that phosphorylates CheY. Now, what would you expect? If CheY phosphate causes the motor to turn clockwise, what would you expect an attractant molecule to do when it binds to the receptor?
It should generate. It should suppress CheY.
Exactly. It should suppress phosphorylation of CheY, and that's exactly what it does. It suppresses phosphorylation of CheY. There's an enzyme that removes the phosphate from CheY. So when you add an attractant, we did experiments where we stimulated single cells with iontophoretic pipettes. We saw within 50 milliseconds after getting an attractant stimulus, the cells started going clockwise. So it's a very quick signal. And it turns out, though, that because E. coli is really small, that the fusion will randomize the distribution of a small molecule like CheY phosphate. It's a small protein. It will randomize its diffusion within 100 milliseconds. So you don't need any way to transport this from the receptors, which are all at one pole, and the flagella, which are all over the surface of the cell. No. If you stop making the CheY phosphate, if you have the enzyme that removes the phosphate, if that balance tips, then the CheY phosphate concentration drops very, very quickly. And the motor is extremely sensitive to CheY phosphate. And this is what the paper from Arvind Samuel's lab in March of this year showed. They could quantify this, and you get a single molecule of CheY phosphate binding to the motor can cause it to start going clockwise. Now, this is really amazing because it turns out there are at least 34 binding sites for CheY phosphate on the motor.
I'm curious, how is it robust to the noise of whatever the bacteria picks up from the external environment?
Well, the noise.
Well, I mean, like the noise in terms of whatever it picks.
Yeah. Well, the noise in the enzyme activity or whatever, right? That's what leads to the running and tumbling. So in order to run and tumble, you have to keep the CheY phosphate concentration in a very narrow range, right? Because if you go a little more of it, they'll only go clockwise. A little less of it, it'll only go counterclockwise. So the whole system has to be designed to make CheY phosphate and break CheY phosphate down to CheY and inorganic phosphate, has to be maintained very, very precisely to get a level of CheY phosphate that allows you to switch between, just stochastically switch between going counterclockwise and clockwise. They have to maintain that in a very, very narrow range, and just a little perturbation of that, right, will cause it to only turn counterclockwise. So you lengthen a run. Or a little increase, you'll get tumble, right? It is very sensitive. Even though there are 34 binding sites, a change of one, right? They don't know what the basic number was, but a change of one is enough to change the direction, to give a detectable change in the frequency.
Wait, so it's not binding to the motor, it's binding, sorry, it's not, is it binding to the stator or the rotor?
It's binding to the rotor.
Okay, so that's why so you can have, doesn't matter how many stators you have.
No.
There's one rotor, so it binds to one of the 34, 35 spots on, and that's
and it must be enough to change the rotor, the 35 thing, from one conformation to another conformation. Mm-hmm. And there's the final trick, right?
But that's interesting because that means the stator is always spinning in the same direction. Is that correct?
Yes, is as long as protons are flowing into the cell,
right? It's always
the
spinning.
stator will be turning.
But if you think of like gears, right? I can't think of any change I can make to the conformation of a gear that would make it go backwards.
Yes.
How does that mechanically work?
Well, the way that works is these guys all spin in the same direction, right?
Yeah.
So one thing I showed way back when with the streptococcus was I could set up a situation in which the proton-motor force was reversed. And I had a mutant that only turned its flagella counterclockwise. I don't know what it was. We didn't have the genetics. But one of those components that makes CheY phosphate was missing, and it only turned its flagella counterclockwise. It couldn't make it. But if you set up a case where now you had a reverse proton motor force, the inside of the cell was positive and the outside negative, then that motor started turning clockwise. So if you reverse the direction of flow of the protons, it became like the ATPase, right? Now you're getting it to rotate in the opposite direction. And I could never understand that. Because in any mechanism we had, were like, you're pushing, when I was teaching this, I used to give the analogy of one of those merry-go-rounds in a playground, where the parents push the thing. And so, if it's very well lubricated, only one parent is enough to push it. But if it's kind of gritty, then it goes faster if you have more parents pushing it, right? And so that's when you would add more Mot proteins on under high load. But I didn't understand how did it make it change direction, right? To go to the other hand, I never could understand that. And I even had worked with a colleague, tried to design experiments because I couldn't see how having a proton coming in from one direction and binding to this site would change the direction that it rotated. Why would it matter which side it came from? In the ATPs, it's obvious because this whole ring of 12 proteins rotates one direction or the other direction, but that's not the case here. Well, it turns out though, if these 5 proteins will rotate, if you rotate them in the other direction, then the motor is going to turn in the opposite direction. So the simple way of thinking of this is, and a picture would probably help, but imagine I have the C ring, this 34 ring, right? And I have this little guy.
That's the rotor.
That's the rotor. That's the thing that connects to the whole whatever, okay? Now, I have these little 5-member rings outside. They're binding to the top of, as that picture shows, they're bound to the top of this ring. This is what's turning. These are the guys that are the stator units, right? Okay? Now, if you imagine it like this, and if this guy's turning outside here, and it's turning clockwise, okay? It's turning like that. What direction is the big ring going to turn?
The opposite direction, let's say.
It's going to turn the opposite direction, right? It's going to turn counterclockwise. What happens if I expand this ring a little bit, and now I have this rotating in here?
Oh, it goes from the outside to the inside.
It goes from the outside to the inside.
Okay.
Now, clockwise rotation is going to drive clockwise.
You don't think of gears as like swelling and passing through each other.
But the thing is, these are proteins, right? And so when CheY phosphate binds, that ring expands. So now these guys are spinning on the inside of it and turning it the opposite direction.
That's crazy.
Is that
so they have to it has to expand. The protein kind of has to jiggle over and sit back down on the inside of it.
No, it stays where it is. So the Mot proteins stay where they are. They're anchored here, right?
Well, it's kind of, I guess so you have the ring, it's on top, it's on the outer perimeter, and then as the ring expands, it kind of
it doesn't move. Yeah, it doesn't move.
This thing binds to the inner perimeter, basically. But it has to unbind, I guess, and then rebind to the inner perimeter.
Well, it's not bound tightly.
Right, it's always going, there's always an on-off.
Yeah.
Wow. Okay.
Is that cool or what?
That's crazy.
Is that...
And so also for this, the proton motors, so they spin, or how does this happen? The conversion of the hydrogen flow into the
hydrogen flow.
Stator.
The hydrogen flow causes the stator ring to spin, and the way it does that, okay. So there are in this center protein. There are, in each one of these, there's an aspartic acid residue right in the middle of the membrane, okay? And that aspartic acid is negatively charged. It prevents the outer ring from rotating, right? So when it's protonated, though, then the ring can turn. There are 2 of them. Now, the fact that there are 5 of these is just, evolution is so fantastic. What is there about 5? Okay. And two of these central ones. How many combinations of positions do you have? You have 5 Mot units. You have 2 of these. That's 10. What's 10 that means 36 degrees, right? Each event, each proton moving through should turn at 36 degrees. And if 10 protons flow through, 5 through one half channel, 5 through the other,
rotation.
then you get 360 degrees. You get a complete turn. So for one turn of this pentameric ring, because there are 2 proton channels, you get 10 protons coming through in the motor turning in 36-degree increments.
So why is it pentameric? Any reason for? It's like just
Well,
the
it could be
structure
different,
of
but
the
it
molecule.
has to be an odd number, right? Because otherwise you get locked in place. It has to have only one of the half channels open at a time. So one half channel has to be open to the outside, the other has to be open to the inside, and you keep alternating between those.
Can you adjust it with mutations? Do like 3, 7?
No.
No.
No, I mean, this is a very precise structure. This pentameric structure is a very precise structure. And as far as we know, every motor for billions of years has been 5-2. I mean, it's the simplest one, right? Well, you don't make things more complicated, right? Like why do we have 13 years periodic cicadas? It's a prime, it's the lowest prime number that's long enough that you can't track it with a predator. Right? So it turns out there are 13-year cicadas and there are 17-year cicadas.
Mm-hmm.
There are no 15-year cicadas. Why? Because 15 is divisible by 5. So you could have a predator with a 5-year cycle that could track it, right? But a 13 or 17-year cycle is just too long for a predator to track it. So, yeah, I mean, evolution knows math, right? Evolution knows math. And it just blows my mind how beautiful this is. You know, I mean, how sophisticated this is. And you would say, okay, and here we get into the philosophical side of it. I don't know if you know, Michael Behe wrote a book called Darwin's Black Box. It's all the things that evolution can't explain, that require intelligent design. Irreducible complexity was his concept, okay? And one of his prime examples was the flagellar motor.
Yeah, I was going to ask, when did the flagellar motor become this poster child for irreducible complexity? Like, when did that all surface?
I don't know if Michael Behe was responsible for it or whatever, but it did. It became the thing, you know, because it was so complex it couldn't. And they said, well, wait a second. No, different components of this exists all over. You know, I mean, there are proton channels, there are things that move, you know. No, you just recruit.
Well, like you said, the 5-2 motor, I guess, is in all sorts of things.
Yeah. Well, you say, well, how did that evolve? Well, it evolved somehow. And then after that had evolved, then, it could be recruited to drive the flagellum, right? So it works. That's the way evolution works, right? It develops one thing for one reason. It doesn't have foresight. It isn't going to say, I want to have a pentameric ring rotating around a dimer so I can drive flagella. No, it made that to do something. And then it turned out, wow, this will be great if I combine it with the rotor, right?
Is there a precursor of the flagellum, which is like a non-moving little whisker or something, like a sensing whisker or something? Or where did the actual like body come from? Of
this whole thing?
Yeah.
Well, there are other things like this, right? There are a whole bunch of type 3 secretion apparati that are very similar to this structure, not the hook and the filament, but this structure, that are involved in, for example, exporting toxins across the cell membrane. And they're very complicated. This structure is quite similar to the flagellum. So there's an export system that works like the flagellar export system, but it's used to export all kinds of other proteins, right? So that is another part. You say, well, how does it export? Well, the export apparatus evolved to export things. And okay, we'll recruit it to make a flagellum, right? So all these things, it was really funny when Michael Behe came to Texas A&M, invited by some Christian group, right? And I don't know how it happened, but he agreed to debate the head of the biology department, who was a friend of mine. So they debated, and Behe had, this fixed thing down that he had. My department head was going through a divorce, and he was not prepared, and he was getting killed, basically. But that he said at one point, he said, actually, there's somebody in the audience that I think Dr. Behe, this is the question period afterwards, somebody in the audience who probably has a question for you or comment for you. And so I got up. And I said, Dr. Behe, are you aware of the fact that the mitochondrial ATPase rotates? Are you aware of the fact that rotation is not reserved to the flagellum? Are you aware of the fact that there's a secretion apparatus that is basically the same as the secretion apparatus at the base of the flagellum? Are you aware that there are these? I didn't know about the 5-2 motors then, but I just pointed all these things that were, and I said, so I said, I'm sorry to say it, Dr. Behe, but you're shooting blanks. You really don't know what you're talking about.
What year was this? Is this like in the 90s? Or?
No, it was in the 2000s.
Oh, okay. So this is relatively recent, actually.
Well, it was after his book was published.
Right.
I said, I've read your book. And so afterwards, it went badly for him then. And the people who'd invited him said, well, Dr. Behe, you're probably tired. You probably want to go to bed. I think they had a party planned, but they canceled it.
How long was the debate?
About an hour.
Oh,
Then with a question period afterwards, right? And then I said, well, Michael, I said, I've read your book, and I know you drink beer. So why don't you come with me over to Northgate, which is where the bars are in College Station. So there are a bunch of biology faculty and philosophy faculty who are gathering over there after the talk. Why don't you come join us? Have a beer, He said, well, will anybody be there that will agree with me? I said, no, nobody will be there that will agree with you, but they'll be friendly. So he came. He came over, and I didn't pay attention while he was there. He was talking to the various faculty and whatever. And then he was staying on campus. So after an hour or two, I said, hey, you want to go back to your place where you're staying? And he said, yeah. As I was walking back, and he said, it was kind of a rough day for you, wasn't it? And he said, yeah, I'm not doing any more debates. He said, I'm just doing my canned presentations. I said, well, so do you still think that you're right about this irreducible complexity? And what he said was so revealing. And there was no witness to this, right? It was just me. He said, first of all, I've got 9 kids that I have to send to college. And, the proceeds from my book and the money I get for going on these things help send them to college. And he said, the other thing is, if I changed my story, the creationists would tear me to pieces. So I said, so you're riding the tiger? He said, yeah, sort of. So I was like, because he didn't really believe it, right? But he is a biochemist. He's a scientist, at least. But he was suborned, basically. He got bribed or whatever. But
do you think when he first wrote the book, he was actually convinced of this, and then basically as he got feedback on the book,
I
he
don't
learned
know.
new things?
We didn't discuss that.
But I guess he wrote the book in the 90s, so maybe also there was just less information.
There was a famous trial in Pennsylvania where they were going to, a school board was going to insist on creation being put into the program, and some students' parents challenged that, and there was actually a trial. And Behe was prime witness for the procreation people. And the guy named Keith Brown, who was I think at Brown University, he wrote one of the 2 books. The Language of God was written by Francis Collins, who was the head of the Human Genome Project, saying that evolution is totally compatible with Christianity because he was a Christian. The other guy, the guy with Brown, was a Catholic, and he wrote another book that said evolution is totally compatible with Christianity. And anyway, he was a witness at this trial, and he just destroyed Behe. I mean, he just absolutely destroyed Behe. And the judge, a conservative Republican judge, at the end of it said, was to be, I think that whoever they were, the plaintiffs, whatever, have made a pretty compelling case that the only reason for teaching creation in school is religious. There's no scientific reason for doing it. And therefore, I don't think it should be done. And so the parents won the case. And
having spent your career looking at these like really surprising things going on, right, at the molecular level, how do you square that? How do you square that with your like religious or philosophical beliefs? Like, does it square for you personally, or?
I don't know if it's all going to be done in the podcast as it appears, but it doesn't matter. I don't care. I grew up as a conservative Baptist. Okay, went to a church where the first pastor of our church was Billy Graham.
Okay.
Wow.
Who went to Wheaton Bible College, which was just up the road from where I grew up in the suburbs of Chicago. And so that's what I grew up with, right? And then as I read more and read more and read more, I said, this was 6,000-year creation is obviously total bullshit. But I still tried to reconcile what I knew about evolution and everything with the religious belief. And here's the critical moment. I don't think my mother will ever forgive my father for this. I had gone to an InterVarsity Christian Fellowship meeting at the University of Illinois over Christmas break on a scholarship from my church. And while there, I devoted myself to the mission field. Okay, I was going to become a missionary. And go out and teach people how evolution was compatible with religious belief or something. I don't know. I get home and I tell my parents over dinner. And my father looks at me and says, "We're sending you to Johns Hopkins so you can become a missionary?" He said, "You could have gone to the Bible college just up the road if you wanted to be a missionary." My mother was staring at him. She could have killed him, right? But I said, they don't believe this shit. My father doesn't believe this shit. Right? So, I mean, that was the beginning of the end of my having any religious belief, because I realized this is all just bullshit stories and just made up. It evolved. Religion evolved just like life evolves, just random variation and natural selection.
But doesn't it bother you that there's something at all instead of nothing? Like, where did all this come from?
Well,
doesn't bother you?
No. How is the idea of God any less absurd than that?
Yeah. It's definitely, at some point they start to blur.
The absurdities, right? The absurdities come together. Yeah, you could have an absurdity that has some great creator, or you could have an absurdity that it just happened, that it was just a big bang. And given the fact that I have no evidence that there's a creator, despite all the anecdotal evidence, all of which can be explained away, it seems the Occam's razor says, no, if you have no evidence that it exists, then why believe it exists?
Do you think it's a good thing to, let's say, this is kind of like the Nietzsche, like God is dead kind of thing, where
no, I don't think God is dead. I don't think God was ever alive. I mean,
this is what I'm talking about.
No, the thing. I mean, God is a creation of humans. I mean, look at God as portrayed in the Bible. Okay, now we're really getting far afield, but he's just a human being.
Yeah. So, but I guess
in fact, he's not even a very nice human being.
We had this kind of helpful fiction in a way, maybe it's hurtful, but like this fiction of around, God and so on. And then science kind of flowered in, let's say, the 17th, 18th century, et cetera. And it kind of killed God in the sense that like the world was infused.
You don't need it.
Right. So this is, do you think something positive was lost through that process, or it's all?
No,
it's all
I think that what was positive about it, I think what's positive about it has become part of human society. I mean, the Ten Commandments are basically maybe the first couple, maybe not, but the Ten Commandments are basically, do not murder, do not commit adultery, do not steal, don't lie. I mean, it's all good stuff. But other religions have done that too, right? I mean, basically the tenets of almost all religions are similar.
So in a way, you're saying that the parts we've co-opted the parts that like similar to using the motor and the flagellum, like the usable parts have moved over. Have moved over.
Precisely, it moved over to governing politics. I think it's really obvious to see how it's developed over. I mean, it's a way of controlling people, right? And as far as we know, the ancient Egyptians, they had a religion. The ancient Babylonians had a religion, but they had multiple gods. Who knows? But it was the same idea. It was a structure to prop up the governing society. And so religion and governance have always worked hand in glove, hand in glove. You had cases of rebellion. You might think, oh, well, Jesus was a rebellion against the order of the time. But how long did that last? How long did Christianity remain a rebellious religion? Not very long.
A few hundred years?
Yeah,
maybe a few, yeah. But, by 300 years after Christ supposedly lived, suddenly that was the dogma. Now you had to believe that. Now that's what supported government. I mean, that's what supported the Roman Empire. Before it was all, the Jupiter and whatever. Now it was God and Jesus who supported the Roman Empire, and it's been very tenacious because it's harder to disprove an invisible God than it is all these guys with all their various foibles and whatever. It's kind of hard to keep believing in Zeus and Hera.
Sure. I would say maybe it's just maybe staying away from maybe religious side, but the question is whether it comes just from evolution, which is a random process, or it's been somehow someone designed it, and then the question is, can we now build this motor? For example,
nobody really designed human society either. It was also just evolved, right?
So, okay, coming all the way back to the story, I think we were in the 80s.
Maybe we'll pass back by.
You guys were working on reversing the direction of the motor. I think that's.
I was marveling that you could reverse the direction of the motor if you reverse the direction of the current, and it turns out if you think this little thing is rotating, the stator, the pentamer is rotating clockwise when protons flow through it from the outside to the inside, then it should rotate counterclockwise when things flow through it from the inside to the outside, and that should turn it in the opposite direction, and that should drive the flagellum in the opposite direction, and that's what happens. Because in the mutant that I looked at, this rotor was stuck in the contracted form, the one that will turn counterclockwise when protons flow in and the stator turns clockwise. What I had done was taken that, couldn't change the circumference of the ring, but if you change the direction of the proton flow, you change the direction and rotate it, right? And I finally understood that. After all that time, when I saw that this thing rotates, I said, that explains it. That explains this mystery that has plagued me the whole time. Why, when you reverse the direction of the proton flow, does the flagellum turn in the opposite direction? Well, it's obvious. If this thing is turning, right? If it turns clockwise, if protons flow out, it should turn counterclockwise when protons flow out. It should drive the motor in the opposite direction if it stays at the same conformation. So all explained.
Mike, engineering question. So what's the efficiency of this motor?
It's remarkably efficient, I think.
And what is the source of losses? Where do we drain?
Well, it's okay. So it's probably not very efficient when the cells are swimming, right? When the fluid turning, it's probably not very efficient. When the tethered, it's turning the tethered cell is very efficient. Sources of loss. God, say I'm not enough of a biophysicist to really tell you what the sources of loss are. But apparently they're relatively. There's very little friction. There's no friction, for example, right?
There's probably still some electromagnetic kind of forces which.
I
mean, friction is an electromagnetic force.
I assume there are. Yeah, I just don't know enough about protein structure and whatever to give you an educated answer to that. But there are a lot of things you have in a normal motor that you don't have to worry about, like friction, right? Or other things that are not a concern at this level, right? Lubrication isn't a concern. So
if
you could approach very, very high efficiency.
So if you stop the flow of the hydrogen, how long will it keep rotating? Would it be like immediately gone?
It'll stop instant.
Yeah, water is very viscous at those
I see.
scales.
It's just
so
like-
Yeah, we
It's
are
just
getting
kidding.
into life and Reynolds number.
So it's just friction of the water, basically.
yeah, which is very
Yeah,
viscous.
yeah,
It's extremely
yes.
viscous at those scales.
So there's something called Reynolds number. So, it's a ratio of inertial to viscous forces. So Ed Purcell, who was at Harvard, when before Harvard went to Boulder, he was before Howard went to Boulder, he was at Harvard. And Ed Purcell was a Nobel Prize-winning physicist there, and he knew about Howard's work. And he gave this talk that has been written down as Life at Low Reynolds number, which explains all of this. It explains how, for a bacterium, it's like a human swimming in tar, right? Inertia means nothing. If it stops actively swimming, it stops within a thousandth of a body length. So it has to keep turning. And the other thing, because there's no inertia, this thing does not work like the propeller on a boat. It's not like it's pushing water. It doesn't push water, right? The reason the turning of the flagella propels a cell is you can understand it if you look at a glass rod, okay? Drop a glass rod in very, very viscous solution, okay? What's it going to do? If it's perfectly perpendicular to gravity, it's going to flow down straight, right? If you put it vertical, it's going to go down straight. At twice, it turns out, for reasons I don't understand, at twice velocity than it will if it's this way. What happens if you put it at an angle like this? It slides. And that's what's happening with the flagellum. Because instead of the thing falling, you're moving the flagellum through a very viscous thing, and that creates a force that pushes it sideways.
Interesting.
And because it's a helix, they add. Like the ones going up and the ones going down add and give a push in the same direction. And it's incredible. I mean, it's just incredible. It's such a clever, you say clever, but it's just evolved, right? It just evolved. That's what turned out to be efficient. And the thing that people don't understand, you don't understand evolution. They don't understand scale. I've thought about teaching a course just on scale. Time, distance, right? How fast do enzymatic reactions? An enzyme can catalyze 1,000 reactions a second. What has to happen for an enzyme to catalyze a reaction? It has to bind things, do something to them, and release them. And it does that 1,000 times a second, an enzyme does, okay? So what's the rate of synthesis of DNA? It's like 1,000 base pairs per second. 1,000 base pairs per second. You synthesize DNA at 1,000 base pairs per second with an error rate initially of 1 in a million. And then with all the proofreading stuff, it comes out to be 1 in 10 billion mistakes that get made. But those 1 in 10 billion mistakes, what are those? Those are mutations, right? And if you do this enough, fast enough, often enough, you get a lot of mutations. They're random. Most of them are bad or neutral, right? Most of them are probably neutral. Some of them are bad. But occasionally, you get one that's good. And you can see that easily if you take, for example, a mutant that has, it's not missing one of these proteins, but it has a defective form of one of these proteins, okay? The one amino acid has changed, so long it doesn't work anymore. And you just put this down into soft agar, where if the cells can swim, they will as they grow, they'll form a ring that grows out, right? Okay, if it can't move, it just stays there, it grows, it's just a blip. You do that, and it turns out sooner or later you start seeing little flares going out that can swim. Why is that? Well, it's either because the original mutation got reversed, Or there was a second mutation that compensated for the mistake of the first one. Well, what are the chances of that? They're very, very low. But there are an awful lot of bacteria there. They're going through a lot of generations. They're going through a lot of replications of their DNA. Where do mutations occur? When you're replicating your DNA, when you make a mistake, right? Well, it's often enough that you get now suddenly, something that's compensated for that.
What are the weakest spots for this mechanism? Like where the errors can accumulate and mutations cause the biggest problems?
A mutation in any one of these components.
Okay.
Any one of those.
But then statistically, which one is the most common? Or like
equally common. It just depends on the size of the protein that's encoded and how critical the structure of that protein is. So, I don't know if you.
Maybe this is like in the context of, let's say, like fertility tests when you look at the sperm cells.
Well, it's really easy. It's really easy to select non-modal mutants, right? I mean, for example, there's a phage. You know what a phage is? A bacterial virus. There's a bacterial virus that binds to the filament and apparently kind of screws its way down the filament to get to the base of that and injects its DNA, right? And it kills the cells. Well, you could easily select non-modal mutants if you just plate them out in a medium that contains this phage, because it'll kill everyone has an operating flagellum, but ones that don't won't be killed. And so you start seeing colonies coming up, and they're non-modal, right? But they'll grow and form a colony. It's because there's a mutation that's knocked out one of these components, right? And so the flagellum no longer works, either it doesn't get made or it doesn't turn or whatever. And there are different mutations that can do that. So
do we know, like, let's say, in regards of like fertility drops, let's say what potentially could cause the problems with motility of, let's say, sperm? Or
what, fertility?
Motility.
The motility of sperm. So the sperm has, I guess the sperm flagella doesn't have exactly the same.
Sperm flagella is utterly different.
Really? Okay.
The sperm flagella, the flagella of eukaryotic organisms are totally, they actually do beat.
Okay.
So they're not rotating.
They don't rotate. They have a totally different mechanism because they're too big. They're too big to rotate.
What do you mean?
The size is too big to get a rotary motor. You start having problems with friction. These rotary motors only work at this size. They don't work bigger. They just don't work bigger. And if you're the size of a sperm, no, you couldn't rotate.
How much bigger is a sperm than an E. coli? It's like a factor of 100?
1,000 times, much bigger.
By volume, you mean something like that?
Yeah, well, I mean, a sperm, I don't know how big a sperm is, but...
I thought like 10 microns? No?
Huh?
10 to 100 microns? Somewhere this case?
The diameter maybe is, like 10 to 100, right?
I mean, you need a pretty good microscope to see it. Like it's...
To see sperm?
Maybe like
nah.
100x?
You don't need it.
100x probably. No?
No, I don't think so. You can see bacteria at 100x. So... I mean, consider, if you start with a single cell of E. coli, okay, and you inoculate it in a growth medium, it'll have a generation time of, say, 30 minutes. How long is it going to take until you reach the saturation, which is a few billion cells per milliliter? It does it overnight, because... If it's 30 seconds per generation,
30 minutes,
24 hours, 24 to the
2 to the 24th.
Yeah, well, actually 2 to the 48th.
Right, 2 to the 48th.
So 2 to the 48th is a pretty big number, and so they grow up and fill the total thing overnight. See, that's why I started working with bacteria rather than with birds. Because you can take a cell and inoculate it, and the next day you have a population of billions that you can experiment with.
What was by the 90s and let's say early 2000s, what were the remaining mysteries in this field? So like, I guess, at least in the Quanta article, you were saying that, okay, this has come full circle. Like we know most of what there is to know now. What was so kind of once a lot of these initial piece of information were known, what were the remaining mysteries for the 90s and 2000? 2000s.
Well, how actually
going into
the motility was generated, how the proton flow through. Well, you knew the protons flowed through this Mot A MotB structure, but we had no idea. We knew that made the protein change shape somehow, but that's all we knew, right? And it was a paper. It was a carefully done biochemistry paper where they tried to work out the stoichiometry of Mot A and MotB. And they concluded, and they might have been biased in their thinking that it should be like this, that it was 4-2. Okay? And maybe because they thought there should be 1 paired with 2, right? Whatever. And so that everybody thought that, okay, it's 4-2. So there are 2 Mot A's for each Mot B. That does not suggest rotation. That suggests there's something, happening there. As soon as the structure came out in 2020 from Nick Taylor's group, and
we only found out 5-2 in 2020.
Yeah,
that's how long it took.
Yeah,
wow.
Because it was not until then did the
microscopes.
Did the cryo-EM get to the point you could look at single particles of that size, right? And so 2 groups, Nick Taylor's group and Susan Lee's group, both came out with structures of the stator unit. So it was missing all this part out here. It was just the part that would be in the membrane and a little bit of MotB that goes through the membrane, right? And it had the little lid thing on it too that I talked about that. But that's what it was. And it came out. It was clearly 5-2. I think everybody, I would guess, everybody who saw that said, "Oh, it's got to rotate." I mean that asymmetry, right? The 5-2, that's got to be important. That's got to be important. And that just, I think everybody started thinking, okay, these things rotate. And in that series of articles that I edited, there's one from Richard Berry's group, and actually he did it with Nick Taylor, who was one of the people that showed that this is a pentameric structure. They propose a model for how it works. They're the ones that proposed that 36 degrees turn. Now, there's nothing about what Arvind Samuel does that says that this works in 36-degree increments, right? All they're seeing is that it rotates. They can't tell what increments it does. But the way they did that, and I'll leave it to Arvind Samuel to explain how they did this, but they could take a cell and tie it down through just a hook, okay? They actually have a sticky hook mutant, so the hook sticks to the surface. And so the cell stays in place. So you can focus on where the motor is, because the motor is right at the base of this flagellum, right? So you can put a little bead on, well, no, you don't even have to do that. No, never mind. You don't need the bead, because you're looking at the rotation of the cell. Okay, so the cell is rotating. Now, but the motor is fixed in place, okay? So now you label the proteins with a fluorophore, something that fluoresces when you excite it at the right length. And here's the thing that I really don't understand, but apparently you can do polarized fluorescence. Okay, so things where you excite with polarized light, and you'll only get a signal if you're in the plane of the polarized light, right? So they took this 5-member guy, they put these fluorophores in all of them. And then this is so clever. I can't believe it. They then just, with an instantaneous, really high signal, they bleached. Really intense signal. And it turns out that you selectively bleach those that are in the plane of that bleaching signal. So if you have a 5-member ring, maybe you'll get 1 or 2 of them that get bleached. Mm-hmm. Now, as this thing rotates, you get fluctuations in the fluorescence signal because you've knocked out some, right? And that's what they did.
Wow. It's like a super resolution approach is like bleach the rest, but leave 1.
Well, yeah, I don't know, 1 or any.
Sure, some fraction.
And And what they could show was that if you had this label on the 5-member protein, you got peaks like
corresponding to 36 degrees, basically.
corresponding to 36 degrees.
But is that basically looking at the rotation?
And it's amazing because if you look at it, and how many were there per rotation? If you took it out over 306 degrees, there were something like 6.8 of the peaks, right? And if you think about this, okay, how many times does a 5-membered ring have to rotate to turn a 34-membered ring through?
Because you're looking at the
306
rotation of the bacteria, not the 5-membered ring, and then you're correlating that to the fluorescent signal from the 5-membered ring.
Yes.
So there's a conversion factor.
And it turns out that if you divide 5 into 34, what do you get? 6.8, which is the number of these fluctuations you would anticipate for one rotation of the bacterial cell. Does that blow your mind or what?
That's
is that
cool experiment.
Is that not clever beyond belief? I mean, that experiment was designed by Howard Berg together with Arvind Samuel. Howard Berg got an NSF fellowship to do that work. He passed away, and Arvind in the physics department at Harvard. So Howard was in the biology department. Arvind was in the physics department. But Arvind had worked with Howard over the years, and Arvind continued on the experiment that they had proposed in this proposal and got it to work. And that's where they published the paper in PNAS. And through some providence, the editor of that paper said, you should get somebody to write a commentary on this. And he suggested me because he said, Manson knows, he was there at the beginning, right? He probably can't tell you how this experiment worked because what does he know? He's not a biophysicist. He doesn't know about polarized fluorescence microscopy, but he can put it in perspective for you and tell you, why this is important, why this is significant. And I ended my commentary by saying, my life's quest is fulfilled. I now, after 50 years, I finally know how what I knew 50 years ago actually works out mechanistically. And that's about as cool as it gets, right? To have that kind of satisfaction, that kind of closure on your career is pretty damn cool.
What are you going to do next?
You have to start a new one.
Next 50 year cycle.
My wife keeps saying, you should write a book about this. You should write a book about this. I say, I'm not a book. I mean, first of all, I think people who write books just have big egos. I don't know. What insight do I have?
Or record a podcast.
Or you do a podcast. And you know what? A podcast is a hell of a lot easier than writing a book.
You can convert it in a book as well.
You go ahead. It's just it's a very interesting story. Just in terms of personalities and everything. And there's a lot of personal stuff that I haven't gotten into. But it's been an interesting ride over 50 years, and I've gotten to meet an awful lot of very interesting people because of it. Some very, very smart people, which has really enriched my life a great deal.
Have people looked at, what's, besides learning the fundamental science of how this works, have people looked at, okay, if we knew how this works, we can make drugs, or we can make machines.
Well,
The
trouble is the scale, right?
But for example, can you kill bacteria now that we know how this works?
Oh well, yeah.
You can say antibiotics.
Well, for example, that but there are lots of ways to kill bacteria, right? So, people say this all the time. Oh yeah, we get a drug that inhibits motility; it'll reduce the infectivity. Well, yeah, but there are a lot of other ways to kill bacteria without And now a big thing is phage therapy, right? Mm-hmm. So because you develop resistance to drugs, the idea is you use bacteriophage to fight the and actually a student that I mentored when I was actually one of Pushkar Leila's students in the chemical engineering department, went to Yale Medical School, and he is working on phage therapy, and he's actually now, I really, this makes me smile, because he said, you've always been so good at trying to make things intelligible to the laity. He said, so I decided to write an article about the phage therapy, and he did that for a journal that apparently is aimed at students. And he wrote an article about the phage therapy, which he did, I think he did extremely well. And he used analogies like, ships going into a harbor and having to find the right dock to get to, like a phage has to find the right receptor on the surface of the bacterium to get in. And of course, you can get resistance to a phage as well. If you just mutate the protein that the phage binds to, then you're resistant. But so you use a cocktail of phages, right? You use multiple phages, and you could do this. Now, is that really better than a drug? You can use a cocktail of drugs too, right? But you can write grants and get money for it. So, which is all that really counts.
Is there a way to make a battery or a fuel cell by running this in reverse? So basically, you spin the motor and generate protons, or something
I don't know.
Have people tried that type of stuff?
Yeah, it's really hard though, because to do this, you need a membrane. It's really hard to create supported membranes. Mm-hmm. Phospholipids. It's basically like a two-headed detergent, mm-hmm. right? And the reason they form bilayers is because you have the hydrophobic tails mm-hmm. sticking together and the hydrophilic head groups, the phospho part, sticking outward. And you make a bilayer membrane, but the most energetically stable form of that is to make a sphere, right? So that presumably in the evolution of life is one of the very first things that happened. You made a phospholipid sphere. Because what you can do if you have a phospholipid sphere is now you can say, I have an inside and I have an outside, right?
Yeah, I
You
guess
have reactions happening inside.
Me and a not me.
Yeah. Your point is that the challenge is that in a battery or something like that, what you really want is a very big flat surface.
Yeah, that's hard.
But you have, like, for example, nanopore DNA sequencers where they will sit a protein into like a nitride or something, like a pore that's drilled in a membrane.
Yeah, I don't say I'm not a biophysicist, so I'm not really the person that could say how this could be used. People obviously try to exploit it, and I gave a talk. For a while I had an Army research grant to work on the Flagellar motor because they were interested in little tiny motors.
Yeah. Did you dream about this, the kind of micro robots, like micro swimmers?
Did I dream about it? No, I didn't.
Making your own ones?
No. I'm not technical. Unlike Howard Berg, I'm not good with machines. I'm not good at any of that stuff. I'm basically a bird watcher, right? I'm fundamentally a bird watcher who's really good at identifying birds. No, and I've tried. I've partnered with a few people. It didn't really get anywhere. When I had this Army grant, I went to a conference at some place near Washington, and there are all these people selling their different ideas. There's certainly a guy there who's doing the ATPase, how you could use the rotating ATPase to do things. My just impression was it was all just bullshit, right? I mean, it seemed to me it was just trying to get money to do something. I think there are better ways to do this. Better ways, microchips. We can do miniaturize things so well without having to mess around with the biology. So I think I don't know, but I'm just dubious about all that stuff. And everybody jumped on it right away. And I remember I was at a meeting in Berkeley. I got to know some people in the chemical engineering department, I think it was, at Berkeley, and they invited me to this conference. And they had all these fancy things they were going to do, right? And then they had one way of sequencing DNA. by pulling the strands apart, right, step by step, and seeing how much energy it required. And they said, a GC base pair is more energetic than an AT base pair by about 50%. So by seeing how much energy it took to pull them apart, you could distinguish. And I said, excuse me, but I have a question. How are you going to tell if it's a GC base pair or a CG base pair? And the guy said, is that important? I said, yeah, that's damned important. So these people are dreaming this shit up and don't know squat, right? So I don't know. I'm very dubious about such things, I must say, which makes me, as a friend of mine who's now, or was at least, the head of the biochemistry department at the University of Washington Medical School, he said, the difference between you and me, Mike, I was thinking I might want to move from Texas A&M, so I was talking about going to maybe his department in Washington. And he said, well, first of all, I could hire 2 young assistant professors for what it would cost to hire you. Which you think is better. And they're going to have 40-year careers, and you're going to have a 5-year career, 10-year career, right? So it's totally uneconomical to hire you. You're not famous enough that, you're going to bring in a whole bunch of money just because you come here. It's not like you're a Nobel Prize winner or anything. And so the other thing is, he says, you're just a damn Luddite. And that's true. I am a damn Luddite. I like experiments you can do with a Petri plate and a toothpick. Those are the kind of experiments I like. I can look at a microscope. I can look at rotating cells in a microscope. I'm okay with that. But all that really highly sophisticated, as I say, I'm still a bird watcher at heart. All that highly sophisticated stuff, yeah, sure. Our lab sequence DNA all the time, mutations, whatever. Did I ever sequence DNA in my life? Maybe a couple of times. Back when you did it all manually, right? And you look for bands on a gel. Yeah, I did some of that in Germany. But have I ever done dideoxynucleotide sequencing? No. Or wait, I'm sorry. That we were doing dideoxy sequencing back then. But anyway, no, I don't know. I'm just not good at that. I enjoy thinking about problems and designing experiments and then having other people do them. Oh, that's what I like. And then looking at their results afterwards and helping them to explain them, right? But yeah, the technical stuff, I'm like the total opposite of Howard that way.
Do you still like to do some hands-on with Petri dish and the toothpick?
No, my hands shake too much. I can't even use the toothpicks anymore because my hands shake too much.
Yeah, so that's the thing. As a biologist, there's a shelf life, right? Because it's very manual.
Yeah.
If you're
it would be like being a surgeon. One time I had a beard, and I thought, I just for old time's sake, I'd like to go and get a shave with a straight razor in a barbershop. And I went in and said, well, most of us don't do that, but whatever his name was, Emil over there, he does that. And whenever I see Emil, and he picked up the razor and he was going like, I said, oh, no, never mind. So,
were there any other?
You need to have the right frequency of the shaking.
Yeah.
Were there any other like really memorable anecdotes in this over the arc of the research around the flagellum? Like anything else that we didn't cover?
Oh, I don't know, probably, but I think we covered most of them. I mean, the really big points to summarize, right? Okay, the flagella rotates. It rotates because these little stator units rotate. You could explain the bidirectional rotation of the flagellum by changing the diameter of the C-ring. The diameter of the C-ring changes because this protein CheY binds to it. And when CheY binds to it in some amount, then it expands to a larger ring. And it goes clockwise, even though the little driving things are still turning clockwise. And then you have the signal from the, and I've worked a lot on the chemotaxis too, on the receptors. Half of my lab worked on the receptors. And so we did a lot of work to show how ligands bind to the receptors and that the signal goes through one subunit of a dimeric receptor and all that kind of stuff. So that's the other big thing, how this gets controlled and how, if you think of a receptor, you bind something way out here, and these are long proteins that go through the membrane as dimers, and the enzyme that phosphorylates CheY binds way down here. So you have to have something, a small molecule binding up here that sends a signal all the way down to here to change the activity of an enzyme that's bound to it.
It's like through membrane, right?
Through the membrane, right? And, that's the whole thing, how that happens. Turns out there's a little push on one of the two subunits, a little bit asymmetric push. There's a conversion device called the HAMP domain at the basis of that. That's a 4-helix bundle that changes its conformation some. That changes the conformation of these long things that go down to the tip. And that changes the activity, changes somehow just the way that the enzyme that phosphorylates CheY binds to that. Its activity changes when this long thing changes its shape a little bit, right? Because, enzymes, that's how enzymes work, right? They have a very specific binding site, and that binding site has to be in just the right orientation for the thing to work. So, if you have it and it's bound in such a way that it's not the right way to do it and you just move it a little bit, now it does it, right? So it can be very subtle changes, extremely subtle changes. You don't see any big thing, any big swing at all. Just very changes of an angstrom or two, but they're enough to change the conformation of the enzyme. So it's either on or it's off. And that signal is carried down through this very long protein with enormous accuracy. It's really cool.
How does it, you mentioned before that, this whole thing has to be kept in a very tight range because otherwise you're not going to be flipping between swimming and changing direction. How does it do that across all kinds of environments, temperatures? Like you would think that even the moment the temperature changes, the whole
now we get into a whole
is going to shoot all over
a whole different thing, which we haven't discussed, is adaptation. So these receptors have glutamic acid residues in them that can be methylated or demethylated, right? And they're the adaptation enzymes. One methylates these glutamic acid residues, the other demethylates them. The key R protein methylates them, the key B protein demethylates them. When the conformation changes, right, when a signal comes in and changes the conformation, it changes the conformation of where these sites are. These sites, because they're charged residues, if they're methylated or demethylated, that affects the conformation of the protein in that area. So, for example, if you add an attractant and you shut off the enzyme that produces CheY phosphate, by methylating some of these glutamic acid residues, change the conformation back to the point where the enzyme is now active again.
Okay?
So you keep adjusting it for the level of, you could have no attractant or you could have saturated with attractant. The cells will still, after an adaptation period, they'll still do the run and tumble, which is important because there are multiple receptors. So if you're saturated with one thing, but you may still be able to detect another thing and respond to that, right? So you have to have an adaptation system to.
So basically, the sensitivity, the dynamic range, you have a bigger dynamic range
enormous.
by tuning through the dynamic.
It's over many orders of magnitude.
Mm-hmm.
And it's readjustable.
And this is that's by tuning the window, basically.
Yes. Mm-hmm.
Okay. And then, but let's say temperature, right? So if I change the temperature,
same thing.
Let's say the enzyme
Temperature changes the conformation of the protein, presumably.
Exactly, yeah.
And then you compensate for that by methylating or demethylating.
who is compensating? Like, whose job is it to compensate? So let's say you stop
The enzymes that methylate or demethylate.
But how do they know? Let's say the temperature is adjusted, and then
They don't know the temperature. The temperature causes
they're
a
just
change.
seeing that there's not
the temperature
enough.
shows a change in the conformation of the protein, and the change conformation of the protein changes its effectiveness as a substrate for the enzymes that methylate or
Right.
demethylate.
Oh, but so it's not that they're reading. So this KY, I think KY protein, the protein that actually talks to the flagellum.
Yeah.
So at some point, let's say you change the temperature, and now there's not enough of it. So there's like no signal at all, and so it's continuously swimming. Is there another protein that notices that and then goes and methylates the receptor, or like?
It notices the conformation change of the receptor
Okay, I see.
that has caused
Yeah.
that to do it, and that change of conformation of the receptor causes it to become
Okay.
methylated or demethylated to compensate. So you're returning back the methylation. I mean, the methylation demethylation is always returning you to the ground state, the state you are when there's no signal at all. It allows you to adapt the conformation of the protein by this methylation to
Reset to zero. It's
reset to
like
zero.
setting
Set the
scale
reset to zero.
Set the
Yeah.
baseline, basically.
Yeah.
Yeah.
Wow.
It's cool.
Yeah. That's.
I mean, I think I made a very fateful and very good decision that night in Charlie Yanofsky's living room, talking to Howard Berg. First of all, I could tell. Well, Charlie Yanofsky is also a brilliant scientist. He's also a member of the National Academy, and he's the one that showed collinearity of the gene and protein. I mean, so he's like done some really, really important stuff in science. But I think it was much more exciting for me to get involved in something where, so in Yanofsky's lab, I only, did experiments and I saw colonies growing on plates. I don't even know if he had a microscope in his lab. He may have had one that he kept in a special drawer that only he could use. So I never saw a bacterium the whole time I was a graduate student. I didn't see bacteria until I went to Howard Berg's lab and started looking at them in the microscope. Because we never looked at a microscope in Yanofsky's lab. We just did molecular biology, right? We just did, and that was because it got me back closer to what I originally was interested in, was behavior. And at one point, when I was a postdoc working with Howard and deciding what I would do, I thought, well, I'm going to go back and see what it would be like to work with birds. So I went to several different labs that studied bird migration. Cornell University has the ornithology lab. That's a big deal there. And this has just come out that birds could sense the Earth's magnetic field. Okay? And there's some really neat experiments done with planetariums showing that the birds would orient to the sky, so the birds that migrate at night. And what they do, they can tell which way is north because of what the sky rotates around. So it rotates around the North Star. If you put them in a planetarium, raise them in a planetarium where you rotate the sky around some other star, they now think that way is north.
Huh.
And so they will, when you do it, they'll orient as though that were north.
You release them into the wild, and they're going the wrong way.
Yeah, they go. Yeah. So, it turns out another thing with birds is that they, a lot of animals, is that they can tell up and down, but they have trouble telling right from left. So they can tell the difference between this and this, but they can tell the difference between this and either that or that. But they have a really hard time telling the difference between that and that. And they're birds they pick up on the West Coast, particularly the Farallon Islands. These are birds that normally migrate out over the Atlantic. Now they went up in the Faroe Islands. How could that be? Well, it turns out if you look from where they breed, it's like 40 degrees one way east of due south or 40 degrees west of due south. So they're birds, they couldn't make the decision between right and left. But once they fly one night going in one direction, they keep going that way. So there's some percentage of the birds that wind up in the wrong place and presumably just fly out over the Pacific Ocean and disappear.
So did you spend some time actually studying birds professionally, or this was
no,
like you just looked into what was the latest?
No, but I like the idea of there was a sun thing, but the magnetic thing with homing pigeons. There was a guy named Bill Keaton at Cornell who studied homing pigeons, and he'd shown you could screw up homing pigeons by putting little magnets.
Yeah, there's some cells with like iron oxide.
Yeah, there are. Yeah. In fact, a colleague of mine at Texas A&M has shown that this is true with butterflies, monarch butterflies. They also use magnetic orientation because they migrate down to Mexico, right? Anyway, I thought, that would be cool. But the experiments in labs had been very inconclusive with, putting birds inside a Helmholtz coil and trying to set up magnetic fields and whatever. And so I had this idea, well, maybe the birds have to be flying in order to either sense the magnetic field or be motivated to respond to the magnetic field. So I had this idea that I would set up a wind tunnel for birds, right? So the birds would be held in place. You'd have the wind blowing, so they'd be held in place. Put that inside a Helmholtz coil, and then have some way of training the birds, like having some wire or something hanging down from their foot, so that you'd give them a shock if they went the wrong way. Okay? So I had this idea. And I went to talk to Don Griffin at Rockefeller University. And Don Griffin's the guy who figured out echolocation in bats. So I thought, okay, he'd be a good guy to do this with. And he said, oh, that's a really interesting idea. I said, well, how would we get started? And I said, well, first thing, you have to find a really big sewer pipe. I said, you mean they don't make wind tunnels for birds?
Can you just study a small bird?
They don't make wind tunnels
Kill
for
them.
birds? He said, no, they don't make wind tunnels for birds. You'd have to make your own wind tunnel for the birds. And then I thought, okay, I could spend a year, and I'm not particularly good at making things. I could spend a year making this, and it turned out the whole idea would be bullshit, right? And I said, okay, I think I'll stick with bacteria. Because that's the problem, right? It's so much more difficult to work with things other than bacteria. I mean, like one of the famous experiments, the Lura-Delbruck experiment, a really famous experiment in biology, it's the simplest thing. This is my kind of experiment. Okay, how do you show that mutations are random? That they're not evoked by the selective stimulus, but they just happen at random, and then the stimulus acts on them. And they did this incredibly simple experiment that was just so conceptually brilliant. Say, okay, you grow up one population of bacteria in one container, right? And then you spread them out on 20 plates and see how many of them have become resistant to a certain phage, okay? And you get numbers of colonies that grow, the ones that are resistant, spread 100 million bacteria on a plate, a few of them are resistant, so you get a few colonies that grow. And it's about the same at all 20 plates, right? And then you do an experiment, you do the same thing, except you have 20 individual cultures, right? In each case, you've inoculated with one bacterium, right? So you have 20 individual cultures. So what happens now? You look at across that, well, most of them have about the same number, but there's some that have no colonies at all. And there's some that have a bunch of colonies. Well, how can you explain that? Well, the only way you can explain that is if the mutation occurred randomly while the cells were growing, right? In some cases, it happened early during the growth of the culture. In some cases, it never happened at all, because the time when the mutation occurs is random. And then they did a bunch of math to show what this meant, but the basic idea is really simple. Mutations happen at random times in the absence of the selective pressure. And that won him a Nobel Prize because it was the first demonstration.
When do you add the phage in that scenario? After you grow the culture?
Yeah, only after you grow the culture. You would just on the plates where you played them out, right? Mm-hmm. So the cells had to have already been resistant before they get exposed to the phage, right? Incredibly simple idea. But I met that guy, Max Delbruck. I've met both Luria and Delbruck. Max Delbruck. So Howard Berg went to Caltech after he was at Boulder, and I moved to Caltech for about a year and a half, which is an amazing place. Caltech is like, I don't know what MIT is like, but Caltech was rarefied atmosphere, right? I mean, you've got Nobel Prize winners coming out the wazoo.
And I guess were you there at the same time as Feynman? Or?
Yes.
Okay, cool.
Yes. And you'd see him at the Athenaeum, which was it's like a faculty club, except everybody could go there, even graduate students. Only undergraduates can't go because they're so uncouth. But graduate students and postdocs, everybody could go. You could sit at a table drinking beer with Richard Feynman at one end of it, just holding court. He was an eccentric sort of fellow, as you know. But he was like, everybody was equal. You just assumed everybody was there for the same reason because they were curious. Exams, you would give exams. My wife and I were resident associates in one of the undergraduate dormitories there for a year. And what a weird collection of people. And most of them socially maladroit beyond belief. And also a ratio of men to women of 6 to 1. So even a girl who'd never gotten any attention in high school was suddenly the focus for all this. And,
Man's monastery, right?
yeah, and of course, totally unprepared for dealing with it. They were all unprepared for dealing with it. And there are all these crises all the time. But the exams, they had a rule. You did not give a timed exam in a room. You handed out the exam. You said, okay, you have an hour or 2 hours, whatever, in these 24 hours to complete the exam. You have to limit yourself to an hour or 2, but you can do it whatever time of day feels best for you to do it, right? And then they had the buildings were open, and when you're done with it, you just put it in a box outside my office, right? Or slip it through a thing into their office. And like, you said, well, would it be cheating? No. It's unthinkable, right? The esprit de corps, that whatever, just the pride, the intellectual pride. No, I wouldn't cheat. It's beneath me. No. That would make me not a Caltech student, right? I mean, it was just amazing to see that, the level of total trust the whole way from beginning freshman students all the way through the faculty. And everybody has kind of respected everybody else. It was really amazing. Stanford wasn't like that. Johns Hopkins wasn't like that.
Do you think it's just the size? Like Caltech is a little smaller, right? I guess.
Well, it's the selectivity. I mean, like, I think, half of the people were valedictorians of their class. I mean, it's a really highly selective group. And they don't select so well. But the thing is, you couldn't flunk out either. So we had guys who were like, in our house, who was taking introductory physics for the 3rd time, right? Because they hadn't passed, but they didn't throw them out. They just say, okay, whatever. It was just amazing. It was just amazing. The students that ran one of the hallways, they were all under 21, of course, but they ran a bar in their room where you could get stuff on credit, and it was just everything. It was just it was the most trusting atmosphere I've ever seen anywhere. And the administration turned a blind eye to all of it, right? I don't know if it's probably not like that anymore, but at that time it was just like everybody trusted everybody else. And it was just...
Did that environment end up producing a lot of
fuck yes
people? I
guess
yes.
So,
right?
Oh yes. It's produced a lot of people. In fact, it produced Howard Berg. Howard Berg was an undergraduate at Caltech.
Yeah.
And as an undergraduate, he worked with Linus Pauling. So, as an undergraduate, you could work with Linus Pauling, Nobel Prize winner. Right?
Do you believe that to be a successful scientist, it's like 95% you have to be in one of these, like in some sort of lineage where like you worked with Berg worked with Pauling, so like your work ended up being important. Like do you think it ends up going
It doesn't
that way?
have to be that way. I mean, there are obviously people that come from modest places, modest backgrounds that have done amazing stuff. I think at some point, though, it's important to get in with people who set an example.
Yeah.
I don't think it doesn't matter where you go as an undergraduate.
It's like a taste of how to do it.
It's a taste, right? So after I was a terrible student, a really terrible student in high school, in college, even in graduate school. I was a terrible student because I was just bored by listening to lectures and whatever. So I just thought, okay, I'll try to get by on just my wits. And it worked just enough. Just enough.
It's a common story.
Just, I had a 2.9 or something like that grade point average at Johns Hopkins, and I got accepted at Stanford. And like, how the hell? Other guys who had, like 3.8, they didn't get accepted at Stanford. How did you get accepted at Stanford? Well, part of it, I guess, was because I worked with Hilary Berger, who had worked with Yanofsky. That was part of it. So there's the lineage coming in. But I'm just also really good at taking standardized tests. So I always thought my intelligence was not meant to get me ahead in the world. It was to make my life easier. So you can say, my mother would say, if you're so damn smart, how come you're not in the National Academy? How come you're not whatever? I said, well, because it's too much work. And also, you have to go to meetings and stuff. It's like, I love to talk, but I hate going to meetings. I hate the stilted talk that takes place at meetings where everybody's trying to impress everybody else with how smart they are, whatever. I hate that shit. And so I would not go to meetings unless it was I had to. So I didn't network or whatever, So you have to network to get into the National Academy, it turns out, So there's a lot of politics to it, and that never interested me. So Howard Berg and Charlie Yanofsky were not networkers. They were just so damn good that they got into the National Academy because they were just so good and had accomplished so much, right? But a lot of people in the National Academy that are,
And what, in terms of, if you were starting out in science today, right? So it's 2026, everything the way it is now.
I'm not even sure I would do it now.
What would you do if you were?
I don't know. I'm not sure I would do it now because, it's so discouraging for science. The atmosphere is so anti-science, so anti-expert.
What would you do instead?
Like, where would you
watch birds? Lead birding tours, maybe.
There's different sides to it. Like, I think there's a lot of amazing things happening, right? So there's
the whole AI thing, which I know nothing about. That's a whole new perspective. And my son got his PhD at Texas A&M in computer science, and he now works for Activision. Mm-hmm. He actually does computer graphics for Call of Duty.
Okay.
And when he got hired by Activision, he said, Mike, I don't know if you're gonna like to hear this or not hear this, but my starting salary is twice what yours is after 30 years at A&M. I said, no, that's great. That's great.
Inflation.
That's not inflation. It's just
with some
things pay better than others, So, that's fun.
But let's say all the kind of like human elements aside, I think just given where the science is today, like where would you go if you were 22 or whatever and you were just starting out? What direction would you take? Would it be biology? Would it be something else? What in biology?
Well, it's hard to say.
Bird watching.
What my curiosity would be. Yeah. As I told you, when I got into what I did through a very unpredictable, uncharted path, right? It just sort of fell into whatever came along as it, but I think what I always looked for was a mentor. Somebody that I could look up to and respect. I remember my freshman high school biology teacher. They called her Mother Nature because she taught evolution. And I remember one time, one of the kids in the class, I was in the honors biology class, although I got such a bad grade that they were going to drop me out of the honors program. But anyway, Mrs. Wenner, I remember some student going up to her and asking her, well, how do you rationalize all this with God and the Bible, whatever? She said, I don't. That's it. I don't. You can't do it. Don't do it. And so they were going to drop me out of the honors program because my grades were not very good. And I got a call that summer from Mrs. Wenner. And this is the first great mentor I've had, okay? And she said, I've told them they should keep you in the honors program. I said, you're obviously bright. If you ever decide what you want to do, you'll do it very well. So they kept me in the honors program. I did okay after that, right? But it was the same way all through my thing. And I'm not boasting. I hope it doesn't sound like I'm bragging, but it's just at each point somebody came in who gave me an example or something. Like, I don't know how I got to do it. I got to go as an undergraduate. Just after I graduated from Johns Hopkins, I got to spend the summer at Woods Hole in the physiology course. I was one of only 2 undergraduates in the course. Other people were graduate students, postdocs, young faculty, right? And I felt totally fine. I didn't feel like I was out of my depth at all, right? Because we were all taking this class, starting at the same place, sort of, and I was as smart as any of them. It was okay. And I remember it was like Caltech, because at Woods Hole, you had all these famous scientists who spend the summer on Cape Cod. And I remember going to the Captain Kidd bar with Seymour Benzer. I don't know if you know of Seymour Benzer, but he's a very, very famous biologist. He was there with Delbruck and Watson and all those guys. And I remember just going to the Captain Kidd bar and just talking to him over a beer, and just his talking to you like you were a normal person. And it was just cool because you could talk to him and you could have ideas and you could exchange it. And so it always just seemed to me that was just totally natural. There was no hierarchy. The hierarchy is all political bullshit, right? It's the mind that where it comes from. And you can have less experience, more experience, but to understand something, if it's within your area that you can grasp, somebody can tell you in 15 minutes or half hour enough that you can reason with them, right?
So now, do you pass this test forward to your mentees?
I try. Oh God, did I? When I taught, yeah. And some students hated me, and some students loved me. I mean, the really good students loved me. The students just wanted to memorize shit, hated me, because I gave them. I asked to teach introductory biology. He says, I'm going to teach this. I'm not going to use a textbook. I'm going to just get a couple of trade books. I got the 8th Day of Creation for them to read. The 8th Day of Creation? It's a story of development of modern molecular biology. It's by Horace Freeland Judson. He does DNA, structure of DNA, the understanding of the genetic code and how genes are expressed, and protein structure. It's those 3 sections. And he follows each one of those through from back at the beginning through wherever they are. And it's great. It tells you how molecular biology developed. I said, okay, student, you're taking cell molecular biology. You should read this. You see how science is done by people, right? And how it happens. And then I got this big, fat book. It's for graduate students, The Molecular Biology of the Cell by Bruce Elberts and some other people. And I said, in this book, okay, it's not a textbook. You could use a textbook, but I just thought each chapter, though, it's so well written. If you start each chapter in a different topic, start reading, just read as far as you can. When you can't understand it anymore, stop. The next course you take, you'll be able to read a little further. And this book you'll have through your graduate school. And by the time you're in graduate school, you'll be able to read to the end of the chapter. So don't sell this book. This is not a book that you sell after you finish the course. In fact, I told the students, I would never assign a book in a course that you would want to sell back at the end of the course. because that book was stupid. It was useless. It was course specific, right? So I use, what are we in what I also taught developmental and evolutionary biology, and then I used The Red Queen, Sex and the Evolution of Human Nature. Some other books about evolution and developmental biology. There were trade books, right? Written by really competent scientists, right? But designed to explain things to the laity. And because it's fun to read. And you really learn something from it, right? A textbook is just deadly. Absolutely deadly. An introductory biology textbook? Good God. It's just horrible. All this crap without any, it's not in context. It's just like, oh.
If there was one, of course, there's so many mysteries left in biology and cell biology and molecular biology. If there was one mystery you'd like to see answered, what is it?
Well, it's impossible. Somebody was asking me the other day. I think it's that, how did life evolve? And could it evolve anywhere else? Right?
It's the origin of life.
Yeah. I mean, if there's life, which almost certainly there's something like life somewhere, is it kind of like life here, or is it totally different? I mean, you still have the same physical and chemical constraints anywhere that life would evolve, right? And, Earth happened to be a very good place for life to evolve because it was the right distance from the right star and spun around at the right rate and had the right amount of water on it and all that kind of stuff. A lot of just fortuitous stuff, but the conditions were such that it was a good place for life to evolve. There must be other planets like that out there, right?
Musical pause.
Keep that in.
Yeah. So to conclude our conversation on the flagellar waters, so is it a completed chapter with?
Oh no! Of
what
course.
is left?
Well, working out the details of how, I mean, there are always more, and who knows? But I think what we know is the basic thing of how it works, right? And now it's going to be refining the assembly process has to be refined. Exactly how when CheY phosphate binds. Why does one CheY phosphate binding enable this ring with 34 members in it to change? How can there be such cooperativity that happens, right? I mean, there's lots of things to work out still. So I don't worry about the people who are working on it having nothing to do. And there's some really bright people working on it. But that's the thing I would say, hang out with the brightest people you can find, right? And learn as much from them as you can. And don't be afraid, because bright people are intolerant of stupidity. They're not intolerant of ignorance, right? And so don't be afraid if you don't know about something, if you ask questions in a way that you can be taught, right? And there's no reason to be afraid. As long as you're sincere, as long as and obviously you have to have a certain native intelligence, but as long as you're sincere about it, don't worry about it. Nobody's going to make fun of you, or if they do, they're assholes, and who cares, right? If somebody puts you off, say, well, screw you. I don't need you. Just assume that people are going to be willing to, if you show an interest and you show some modicum of ability, anybody who's worth their salt as an academician is going to spend time with you. And the ones that won't are just jerks.
Great. Well, that's a great
Thank
ending.
you for your time, Mike.
Thank you, Mike.
Yeah.
Okay.
Mike, thanks again for taking the time
You're
to
welcome.
do the interview. We were able to print a 3D model of this whole motor complex
I
that
see
we
that, yes.
discussed. And so we'd really like to take our audience through a lot of the 3D parts because in the video, I was able to put in some images and animations, but I think there's also something valuable to having the tactile experience of being able to point to the individual
Okay,
parts.
so what I will try to do here is to reconstruct this. Now, this is not going to be reconstructed in the way that this assembles inside the cell, right? So the core, the first thing to form that you can detect as a flagellar structure is this ring, the MS ring, and this is embedded in the cell membrane. Okay, and it's the foundation on which the whole rest of the flagellum is developed, and it has a number of subunits. It forms a ring, and this is a fixed structure. This is always going to be the same, and it's what connects what's inside the cell to what's outside the cell. So that the rotation of something inside the cell can cause the rotation of something outside the cell. Okay? So this is the platform for assembly. So what assembles on this are two different things. And I'll show first the external part of this, because this is what was first discovered. It was easy to isolate. You could isolate the external part of the flagellum as a separate structure. And so what goes inside of this MS ring is something called the rod. Okay, and it attaches to, if I can balance that there, that sits like that in the MS ring. And it assembles on the MS ring. Okay, and this is outside the cell now. So the basic part of it, so E. coli is a gram-negative bacterium, which means it has a cell membrane, it has the peptidoglycan cell wall, which is relatively rigid, and then it has an outer membrane also, which is fluid. So this has to go through the space called the periplasmic space, which is between the cell membrane and the cell wall. It has to go through the cell wall, and then it has to go through this outer membrane. Okay? And the way it does that is there's another ring here. If I can get it to fit on there or not, let me show it. So I'll bring it up from inside. So this ring is called the LP ring, and it's not going to quite be able to fit through here. But this ring goes inside this LP ring. The LP ring, the P part of it is the protein that associates with the peptidoglycan. The L part of it is the part of it that associates with the lipopolysaccharide outer membrane. So this is a bushing, basically. This does not rotate. The rod goes up through the middle of that and rotates. Okay? And now I can't push it through far enough to make it work, but you just have to imagine this is rotating inside this bushing, which allows the flagellum to penetrate the cell wall and the outer membrane, and then assembled onto this. Actually, it's not assembled onto the bushing, it's assembled onto the tip of the rod, is the part of the flagellum that's outside the cell. And so if you look at an image of it, the part I'm holding in my hand here is the cell proximal part of the flexible hook that's at the base of the flagellum. This hook goes out, it's 55 nanometers in length. It's always that same length. And when the hook reaches that length, there's an amazing process that occurs. There's a signal sent back through the whole thing, back down to an export apparatus. And the export apparatus is assembled on the inside of the MS ring. It would be down here inside the cell, right? And that export apparatus exports all the proteins of the rod, the hook, and then eventually the helical flagellar filament, which is attached to the distal end of the rod. Okay? So you have a picture there of what the entire complete structure looks like. But what you have to realize that all of this rotates, right? All of this rotates. The MS ring rotates, the rod rotates, and the hook rotates. And because the filament is attached to the hook, the flagellar filament, the helical flagellar filament also rotates, right? And because the hook is a flexible structure, it allows a rotation that's perpendicular to the cell wall to bend around 90 degrees. And there are multiple of these flagella per cell. They can all bend around 90 degrees, and the helical filaments come together in a bundle, a helical bundle, and it's the rotation of that bundle that actually drives the cell. Okay?
I see. So each of these motors, there might be multiple.
In E. coli, there are 3 to 6 of them per cell.
I see, and they're all localized on.
No, they're random on the
see.
surface.
Okay.
Right?
Yeah,
But because this hook allows them to bend around 90 degrees, they can all come into register to form
so they can
a
orient
helical
themselves.
bundles.
I
Yes.
see. Interesting. That's very
Okay.
cool. So it's not just one bacterial cell with, like a boat propeller on the back. You've got all these.
Right. There are bacteria that are like that.
Sure.
Right. But
Yeah.
E. coli is not like that.
Yeah.
It's called peritrichous, meaning all over flagellation, all over hairs. I think peritrichous means all over hairs or peripheral hairs. And there are a lot of bacteria that are like that. There are the bacteria that have polar flagella. There's all kinds of different patterns of flagellum. In fact, there are even bacteria that have internal flagella
Wow.
called spirochetes.
And so are these pointing into the cytoplasm?
No, they point into the space between the cell wall and between the cell membrane and the cell wall. They rotate also, but what their rotation does is it drives the rotation of the entire cell body. The entire cell body is spiral, is a helix, and so the rotation of the flagella inside the cell causes the entire cell body to turn, which then spirals its way through the medium. And this is something you can imagine in an environment that's very viscous, where you don't want to have these thin filaments that can get all tangled up. If you keep the filaments inside the cell body, you can go through a much more viscous environment if the whole cell body is moving and they just sort of screw their way through. So
Turns
spirochetes.
itself into like a little bullet.
Yeah, but it's the same. It's the same mechanism. The motor is basically the same. It's just where the filaments either go outside the cell to form a structure that drives the cell, or they stay inside the cell and cause the entire cell body, which is a helix, to rotate. So that's important. That fact it's a helix is important for the mechanism of propulsion. Okay. So now what I've described is all what you would see outside the cell. But it turns out the really interesting part of it, the motive part of it, is inside the cell. It's in the cytoplasm of the cell. And the MS ring is assembled, or the ring inside assembles around the MS ring also. So everything outside assembles around the MS ring, but what I'm about to show you also assembles around the MS ring. And that's called the C ring. The C ring is way bigger, right? Here's the MS ring, and that fits inside of here and attaches to this, okay? But it's a firm attachment. So if this rotates, this is going to rotate, and in the same direction. Okay. Now, this ring consists of 3 proteins: the Flag protein, the Flag protein, and the Flag protein. It turns out the Flag and Flag are important because they are what control the direction in which this rotates. Okay? They're not involved in actually the propulsive mechanism, but they're what controls the direction that this goes in. Okay? And as you can see here, there are 2 different, these are both the C ring. They're 2 different conformations. And we'll get to what these little things on the outside are. So these protrusions that are sticking up in either of these models are not part of the C ring. The C ring is that ring without these things on it, okay? And the flagellum is made like that, with the C ring attached to the MS ring, attached to the rod, going through the LP ring bushing, connected to the hook, connected to the filament, okay? But all of that, when the C ring rotates, the MS ring rotates, the MS ring rotates, the rod rotates, the hook rotates, the flagellum filament rotates, okay? Now, that's all fine. But what you can see is that this C ring can exist in 2 different conformations. There's let's say a condensed formation, which is here, and an expanded. And I think you can see that the diameters are, maybe you can see it, are a little bit different, right? So one is just a little bit higher diameter than the other. This one has a little bit larger diameter, okay? So that's important because these two different conformations of the C-ring correspond to the two different directions in which the flagellum can rotate. So the way E. coli swims is that it has left-handed helical flagellar filaments. If those filaments are rotated at their base in a counterclockwise direction, the filaments come together, form a helical bundle that pushes the cell along. If the flagellum starts to rotate clockwise, that bundle falls apart, and the cell goes through a chaotic motion known as a tumble. So the basic behavior of the cell is to swim with counterclockwise flagellar rotation in a straight line, and then tumble when the flagella start going clockwise, and then going back to counterclockwise rotation, the cell takes off in a new direction. So what the cell does is a three-dimensional random walk, and the reason they do that is so they can do chemotaxis. And the way they do chemotaxis is by biasing the random walk and making the runs with counterclockwise flagellar rotation longer when it's going in the direction that's good for the cell, when it's going towards a nutrient or away from a repellent. And the cells can't swim for a very long time in a straight line. They can't home in on a signal. So eventually they're going to go off path. Then they're going to stop having an increase in the concentration of the attractant chemical, and then they'll tumble. They'll go off in a new random direction. If it's not a good direction, it won't go very long. But if it's in the right direction, then they'll go for a longer time. So the random walk is biased. And that's how they actually move in a gradient, by going for a longer time in a positive direction. And Howard Berg said that means, bacteria are optimists. That if things are getting worse, they ignore it. They just keep doing what they were doing. If things are getting better, they go longer in the direction which is getting better.
Are you suggesting then that they only behave in a positive direction, meaning they'll ascend whatever gradient, but not descend away from a repellent,
they will.
for
They'll
example?
descend a repellent gradient,
Okay.
because then they'll lengthen their runs when they're going down the gradient.
I see. Okay.
Okay.
Yeah.
So it's the same mechanism. They bias to go the direction they want to go.
Right. Okay. I see.
It could be up an attraction gradient or down a repellent gradient.
Okay.
But it's the same mechanism. It's
Yeah.
a biased random walk, right? Okay. So what are these little things stuck out here? Well, it turns out that let's see if I can find the. Oh, here it is. Yes, and if you can focus in on that. Okay, that's a single one of these things that you see attached around here. These are made separately from the flagellum, and these are a structure that consists of two proteins. There are two copies of one protein, MotB. There are 5 copies of the other protein, which is MotA. Okay? Now, this little thing is what generates the motility. Because it turns out this little thing, which penetrates the cell membrane, and this is, could think of this. So if I hold it like this part up here is outside the cell membrane. The bottom part here is inside the cell membrane, and this part in the middle penetrates through the membrane. Okay?
Right where those two little legs are?
The two little legs are outside.
I see.
This part here is in the membrane, and then the bottom part,
Got it.
the pentamer down here, is inside, right? So there's a structure that goes through the membrane. Now, the critical thing is that this part out here is what attaches to the cell wall. If you think you're going to rotate something, a rotary motor has to have a rotor and a stator, right? Okay? This is called the stator unit. And the reason it's called the stator unit is because this part out here attaches to the rigid cell wall. So it forms a fixed attachment against which the rotation can be made. Okay? Now, this protein is made separately from the flagellum. And it just floats around in the cell membrane. And when it's floating around the cell membrane, this part that attaches to the cell wall is compacted. So it doesn't attach to the cell wall. It just floats around in the membrane. Only when it hits the C ring does it go through a conformational change that extends this part so it can attach to the cell wall, right?
See.
The other thing that does, that process of that conformational change that allows us to attach to the cell wall, opens up a channel, an ion channel in this structure that allows ions to flow through. And it's that ion flow that actually powers the motility. In a way, I'll explain in a moment. Okay, so this looks like it might be a fixed structure, but it's not. There can be anywhere from 1 to up to 10 or 11 of these things attached to the C ring, these MotA B complexes. Okay, and the complex, as I said, is 2 MotB proteins, the things that go out and attach to the cell wall, and 5 MotA proteins. Okay, now MotB attached to the cell wall. What does MotA do? Well, MotA is what interacts with the C ring. Okay, and there are electrically charged residues of opposite charge, so there's electrostatic interaction between the MotA protein and the Flag protein of the C ring. And here's the amazing thing that came out just within the last year. Okay, so the structure of this unit, the stator unit, was determined by 2 different labs in 2020. Nick Taylor's lab and Susan Lee's lab. Okay, and what was amazing about it was everybody had thought that the stoichiometry of this was 4 MotAs to 2 MotBs, but it turns out the real structure was 5 MotAs to 2 MotBs, which seemed weird, right? But right away, it started getting people to think, geez, maybe this is a rotary structure too, okay? And it turns out it is. So what happens is, the way this works, is it's floating around in the membrane. It's not conducting any ions. It hits the C ring. It undergoes a conformational change in which this MotB part that's outside the cell extends and attaches to the cell wall, and at the same time that opens up the ion channel. The ion channel is closed when they're just floating around the membrane. When it hits the ring, the ion channel opens and it attaches to the wall. Now, the thing is that the pentamer here, as ions flow, and it can be in some bacteria, it's a sodium ion. In E. coli, it's a hydrogen ion, either as a proton or as a hydronium ion, H3O+. And that ion has to flow through the membrane, through an interface of the MotB protein, which penetrates through the membrane, and the MotA protein. Now, if you think about it, okay, the stoichiometry of that, there are 2 MotBs, there are 5 MotAs. Each MotB protein has a charged residue, an aspartic acid residue, that is absolutely critical for motility, because it turns out that the positively charged ion comes in and binds to that charged residue. The charged residue prevents the thing from rotating, but when something binds to that neutralizes that charge, then it can spin, right? And the way this is set up, this proton channel is set up, or ion conducting channel is set up, is a half channel. One part of it extends out to the outside of the cell, or the periplasmic space in the case of E. coli. And the other half channel, which is offset by 36 degrees, goes into the cell. So how can an ion go through this channel? Well, it has to come in through the outer half, bind to the aspartic acid that neutralizes that charge, so that the Mot A pentamer can turn 36 degrees until it releases the ion into the cell.
I see.
And if you think about it, the stoichiometry of it, there are 2 Mot Bs, there are 5 Mot As. The channel is made up partly of Mot B, partly of Mot A. The channel through the membrane can only be created by rotation of the Mot A pentamer. And so per 360-degree rotation of this structure, there are 10 protons that go through. Okay? And each proton that goes through causes a 36-degree angle turn, right?
Wow.
And the 2 half channels are set off by 72 degrees. So halfway gets you go half of the 72 degrees, and then you go the other way, get rid of it, and you go the other half, okay? So, and that happens 10 times. 10 protons go through per rotation of this guy. Well, what does the rotation of this guy do? Well, remember, this is attached to the cell wall, but this bottom part is rotating, right? Well, this bottom part is in contact with the C ring. And as unlikely as it may seem, even one of these guys can turn this whole C ring. If the C ring turns, the MS ring turns, the MS ring turns, the rod turns, the rod turns, the hook turns, the hook turns, the filament turns. So one little thing like this, conducting protons, down here attached to the C ring, can drive the rotation of the flagellum at hundreds of revolutions per second.
Wow. Just this tiny protein can spin this.
Yes.
This big ring.
Tiny protein is spinning this big ring. And if it takes 10 protons per revolution of this, now consider the stoichiometry. This is a pentamer, right? This is a 34 subunit ring. So that means that you need 5 divided into 34. You need, in principle, 6.8 rotations of this to get one rotation of this big ring, right? But one of these is enough.
Wow.
To drive the rotation of that whole thing. Okay, that's a bit at 300 revolutions per second.
Yeah.
Now this shows a whole bunch of them, and that's because when the cell is just swimming freely in water or a non-viscous medium, just one of these is enough to drive it. But if it gets more and more viscous, there's some more resistance to the flow. It needs more of these units, and so up to 10 can be added in when the motor is operating at really a high load.
Yeah.
Okay, now the other part of this that's important is seeing the difference between these 2 conformations. So both of these have the full complement of Mot AB attached to them. But what you see has happened. Now remember, the Mot AB are attached to the cell wall. They're fixed in place. They can't move very far. Just whatever flexibility there is in that linkage. What happens, and here's the amazing thing, is that the diameter of the C ring changes for the 2 different directions of rotation. If you're in the closed conformation of the C ring, the Mot B, Mot A stator unit is outside of the ring, right? So what's going to happen? It's just like gears. If this is outside and it's turning clockwise, and it turns out that it has to turn clockwise to get the proton to go through, when the proton comes from the outside to the inside, then if it's on the outside, as this turns clockwise, the C ring is going to turn counterclockwise. Okay? So how does it change direction? Well, it changes direction by changing the diameter of the C ring.
And that's because the Mot A, Mot B can't itself
That can't move.
change the direction.
So it
of
has to
spinning.
change the ring, right? And now here you have the Mot A B units on the inside of the ring, and now when they turn clockwise, the ring is going to turn clockwise. right? You can see that simply if you just draw it as gears, So why does that happen? Well, it turns out, I don't know if they have it here, it's a tiny little protein, but a tiny little protein binds to the Fly-M and Fly-N proteins I told you are the bottom part of this C ring. And when the CheY protein binds to that, it causes the C ring to go from the constricted or contracted formation that causes counterclockwise rotation, which causes smooth swimming, to going to clockwise rotation, which causes the tumble. Okay? And it's just by expanding the diameter of the C ring. The Mot A, MotB stator units are always turning clockwise, but depending on how they interact with the C ring can turn clockwise or counterclockwise. And one of the discoveries I made when I was a graduate student, like 50 years ago, or a postdoc 50 years ago, was astonishing to me, and I could never explain it until I knew that these stator units rotated. Which was that I had a cell which was a mutant, and it could never turn its flagella clockwise. It always swam smoothly, right? It always had counterclockwise flagella rotation. And I could set up, with the particular bacterium I was using, I could set up artificial gradients across the membrane. So normally, the inside of the cell is negatively charged with respect to the outside. So a positively charged ion, like a hydronium ion or sodium ion, is going to flow in, okay, in response to that electrical charge. And it turns out, if a hydrogen ion, it also can be a pH gradient. It's going to be the hydrogen ion concentration that drives the thing, if the inside is alkyl in respect to the outside. Now, what I could do, though, with this particular bacterium is I could create a situation in which I could artificially reverse the sign of the proton motive force, okay? Normally, the inside of the cell is negative, the outside is positive. I could manipulate it so the inside was positive and the outside was negative. And when I did that, lo and behold, this cell that could only turn its flagella counterclockwise turned its flagella clockwise. And I could never understand that.
Wow.
But now I do. Because if you consider, if these units are always turning, the stator units, the MotB, Mot A pentamer, are always turning clockwise. If protons are flowing into the cell, if protons are flowing out of the cell, it has to turn counterclockwise. And if you have something that's stuck in this conformation, always, where it can only turn counterclockwise,
Right.
and you flow the proteins the other direction, and this starts turning counterclockwise, lo and behold, the ring is going to turn clockwise, right?
That's amazing. So the whole structure, in a way, is reversible in two separate ways.
Yes.
Either by changing the conformation of the ring with the Mot A, MotB in the same direction,
Or changing the
or
direction.
locking the.
But of course, the
That would be in a mutant.
well, it's not, I mean, it would never happen. You would never, under normal circumstances, you'd never have a cell that's positively charged inside
Right.
and negative outside, because
Yeah.
it's non-viable. You wouldn't, but it can endure it for a little bit in an experiment. And when you create that condition, you can see the result. And I could never explain that. But now that we know that this unit rotates, it's simple to say, if the flow goes the other direction, the stator unit is going to rotate in the opposite direction, and it's going to turn the flagellum in the opposite direction. So that's why I said, so how I got into this recently was by reviewing a paper from a successor to Howard Berg, R.V. Samuel at Harvard, and he and Howard Berg designed an experiment in which they could actually look to see if these units rotated. It's a very sophisticated thing. I can't really explain it. It involves some very sophisticated polarized fluorescence microscopy. But by using this and having fluorescent labels on the MotA protein, they could actually show in a cell that was held in place in space, they could show that the fluorescent things attached to the MotA protein rotated. And so that was actually a physical demonstration that this tiny little thing, which you could never see under normal circumstances, right? This is like, 10 angstroms in diameter or something like that. Not 10 angstroms, 10 nanometers in diameter or less. You could never see that with any kind of normal microscopy. But with this polarized fluorescence microscopy, they could actually see a pulsatile signal in the fluorescence that was explained by the rotation of the seed ring. And it turned out the amazing thing was they could simultaneously record the rotation of the cell and the rotation of the seed ring. And it turned out, wow, there were about 6.5 of these pulses per rotation of the flagellum. And okay, what do you get? Well, there are 5 subunits here. If each one of those 5 subunits interacts with one of the Flag subunits as it turns, well, it should take 5 divided into 34, 6.8 rotations of the little guy to drive one, and that's exactly what they saw.
Yeah, it's a really, really beautiful and elegant experiment. Manson explains it in a little more detail in the full interview, so definitely check that out if you're interested.
But anyway, I just think it's absolutely amazing. And as I said in the commentary I wrote for the Proceedings of the National Academy of Science, which I was commenting on the paper from R.V. Samuel's lab in which he did this fluorescence microscopy, that it was like the fulfillment of my career. I've been working on this. The first paper I published on this, when I showed that it was actually a flow of protons that drove the rotation, was in 1977 in PNAS. And now, 49 years later, also in PNAS, is a paper that shows how this thing works. How the proton current actually drives the rotation of the flagellum. So for me, I was like thinking, I'm done. It's not like there's not more to learn, obviously.
Yeah.
But it's so satisfying to see your kind of what you've been working on for your entire career come to fruition. And now I understand how it works. Wow, that's so cool.
That's kind of the whole impetus for doing science.
Yeah, how often you get there.
And very few scientists can say that. So
yeah, I mean, it's almost like, Watson and Crick saying, oh, wow, it's an antiparallel double helix. I mean, that explains everything. That, what is that? That explains how DNA replicates. That explains everything, right? I mean, it's so, once you know that, there's just these. Now, I'm not saying this is of the same magnitude necessarily as the structure of DNA, but it's just telling you how you can make a molecular motor and how all these different parts are seen in other contexts in cells. And so what the flagellum is kind of an amalgam of components that have come together to make this machine. For example, the unit that exports the proteins, that all the proteins that are in the rod, the hook, the filament, have to be exported out of the cell. They're made, everything's made inside the cell, right? They have to be exported. And they have to be exported in the right order. Well, it turns out that there are structures in bacteria that secrete toxins out of the cell. Their export structure is the same as that of the flagellum, basically.
And when you mean the same, are you implying any sort of homology or just
structurally
mechanically very similar?
Well, structurally similar, yes. And it may be evolutionarily of a common origin, right?
Yeah. I mean, especially if these same gears within this structure are repurposed or remodeled in certain ways.
Yeah, but I don't think the export structure in the secretion of toxins rotates.
Sure.
But it has to export things, right?
Yeah.
And the flagellum has to export things. And you can bring in other things. These little stator units, turns out, show up in other contexts in the cell. They're part of other things where the cell has to do work outside of the cell membrane. These are attached to things that are involved in the synthesis of the cell wall. These are involved in things that are involved in transporting things through the outer membrane of the bacteria, and they have these 5+2 motors. Now they're saying, oh, these 5+2 motors are everywhere, right? And they're in all kinds of contexts that have nothing to do with flagellar motility.
Right.
And so things get borrowed, right?
Yeah. I mean, like everything in biology.
And the other amazing is rotation. I think I mentioned that I edited a series of 20 papers talking about biological rotary nanomotors. They're all over the place. And they're these 5+2 motors, but it turns out the ATPs you have in your mitochondria that every cell has, it's also a rotary motor that makes ATP, right? And it's just these basic principles. And the cool thing is that if you're at the molecular scale, you can make motors that don't have the problems of bigger motors. They don't have to worry about friction. You don't need a lubricant. So rotation can work in a molecular motor without a lot of the problems you get if you try to make larger motors.
You get a lot of benefits of being small.
You get a lot of benefits of being small. And I quickly concluded this. It turns out that this is probably the only way you could swim if you're really small. You couldn't swim like a fish. You could, reciprocal motion doesn't get you anywhere. Because it's the Reynolds number, right?
Yep.
Ratio of inertial to viscous forces, right? If you're very small, inertial forces are negligible. It's all viscous forces, right?
Yeah.
So anything that involves inertia to drive it, like a propeller of a ship pushing out water and counter-reaction to that is pushing the ship forward, it doesn't work, right? Because you don't push water when you spin. What you do when you spin something is you glide through it, right?
It's almost more similar to a burrowing mechanism than it is to swimming. So
yeah, it is.
Yeah.
Right? It's not. So that doesn't work. It's called the scallop theorem in this paper by Ed Purcell, Life at Low Reynolds Number. He shows the scallop theorem. He says if you were a scallop the size of a bacterium, how does a scallop work? It closes its shell quickly and then it opens it slowly.
Yeah.
Well, that works. If you're big, well, scallop is big in the terms we're talking about. If you're a bacterium and you did that, you'd close it, you go forward, you'd open it, and you'd go back the same distance. Because there's no inertia, right? So you would just go backwards and forwards. You wouldn't get anywhere. But if you're revolving something, then you can actually generate a force that pushes you along.
Amazing.
Not because you're pushing water back like a propeller, because you're gliding
drilling
through it.
into it.
Yeah.
Yeah.
So that's the story of it, really. Really amazing. And then you take over, you put in the chemotaxis. Okay, you have receptors that sense the chemicals, and they send signals to the motor, and all the signal is this little protein, CheY. CheY, when it's phosphorylated, binds to the motor and makes it turn clockwise, makes it tumble, right? So how do you go up a gradient, make your runs longer? You inhibit the synthesis of CheY, phosphate, right? You don't phosphorylate CheY. You don't phosphorylate CheY. It doesn't bind to the motor. The motor doesn't go clockwise, and you swim longer, right?
So I have a question regarding that CheY mechanism. So, and I apologize if you covered this in the interview. If you did, I'll just cut this question out. So when the CheY binds to that ring, in order for this biased random walk to happen, it must then unbind, or in some way deteriorate.
It has to be going on and off, on and off.
So,
in order for the cell to do the random walk, right? CheY phosphate has to be binding when it tumbles and unbound when it doesn't. Now, we don't know. In principle, there are 34 places for CheY phosphate to bind. The recent study, also from the Samuel lab, showed that just one CheY phosphate binding to the motor is enough to switch the direction from counterclockwise to clockwise.
Right.
Now, we don't know. it does. You don't know if that's going from 0 to 1 or from
but the delta of
one
QY.
is enough to change the direction. So it has to be extremely finely biased, right?
Yeah.
And that's why obviously the level of CheY phosphate in the cell has to be maintained very, very particularly in order to get the run and tumble behavior, right? And if I add a lot of attractant and I shut off the synthesis of CheY phosphate, the cells will swim smoothly for 10 minutes or something. They'll eventually adapt because you change the receptor so it no longer inhibits the phosphorylation of CheY. But for a long time, you'll just swim straight. But of course, that's useless for the cell because the cell can't really swim straight. It is always going to curve, and it can't go up the grade. It's going to curve off and go somewhere, right? So it has to constantly reset its direction by tumbling. So it can sample a new direction, and if it's a good, keep going a little bit longer. If it's not good, tumble again.
Yeah, it's amazing. Do we have any insight on how this CheY unbinds? Does it get spent as the motor spins? Does it just
there's a protein, essential protein called CheY, which binds the CheY and removes the phosphate.
I see. Okay. So that's another thing that comes in and
so
peels it off.
once you stop making CheY phosphate at the receptor patch, very rapidly the concentration falls because this CheY removes the phosphate. So you get some experiments I did in Berg's lab way back when showed that the cell responds, the motor responds to an attractant stimulus. Even so, the receptors are all one pole of the cell, and the flagella are all over the place, right? But even in a flagellum, which is far away in terms of the bacteria, which means like a micrometer, from the time you add the attractant until this starts going counterclockwise exclusively is like 50 milliseconds.
Yeah, because just diffusion alone can.
But the fusion is so fast at the scale of a bacterium. The fusion is so fast that if you stop making CheY phosphate over here, the concentration falls because of CheY. The concentration falls below the threshold for getting clockwise rotation, and then you keep going smoothly.
Wow.
And and that system, the whole thing. I say the receptors receive a signal, they send a signal. Oh, you've seen an attractant, but they adapt. So that the fact that they adapt means you don't just keep swimming straight all the time. You get a certain increase in concentration, and then you adapt to that, and now you're reset, and now you can detect further changes, right? So you can move up a gradient by constantly resetting the receptor. And through the adaptation of the receptor, and so you can go up many orders of magnitude of concentration gradient because you could keep resetting the receptor.
Right, you can start shifting your window of relative what's in a positive or a negative ascent.
But you have to still keep the CheY phosphate level has to stay at the same level.
You can't just fill the whole cytoplasm.
No. So you have to keep, what the modification of the receptors does is change their sensitivity to the attractant, right? So they get less and less sensitive. As you get more and more of the stuff on there, you get less and less sensitive. And
it's mind blowing to learn about the literal behavior of these cells, because typically people don't think of anything as really behaving that doesn't have neurons, for example.
Yeah, right? Well, this is behavior, and it doesn't involve neurons. But I mean, just think about it. Like one of these little guys, right? One of these little guys turning can drive the rotation of this whole thing: the C-ring, the rod, the hook, the filament. One of these guys can drive that at 300 revolutions per second. Now, what's the ratio of the weight of the motor of your car to the total weight of your car?
It's almost like 50%.
Well, it's that high, but it's a lot, right?
A lot, yeah.
What's the ratio of this to this other structure?
Yeah, that's like a couple Daltons or something, or?
Oh no, there's many Daltons, but I mean it's like, probably. So the MotB protein is 30,000 Daltons, and the Mot A is maybe 50,000 Daltons, and there are 5 of those. Okay, add it up, blah 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 blah. But it's trivial compared to the rest of the thing, right?
Wow.
So it's an incredibly efficient motor. Incredibly efficient motor. And people say, "Oh God," as I said when I wrote my commentary, somehow this got picked up by Quanta magazine, and they wrote an article about it. They had a Zoom conference with me, and then the Quanta article was read by somebody in an automotive magazine, who then said, "Wow, if we could only make cars that had motors like this, right? That would be so cool." But there are problems. Things don't scale particularly well, right? There are things you can do on the nano scale that you can't do on a bigger scale.
Yeah.
So
One thing that, we've discovered over the past, 40 plus years is that they're more ubiquitous than you might intuitively think some complex structure to be, right? So we're finding them not just in the flagella, but like you said, ATP synthase, there's other ones.
Yeah. They're basic physical principles that constrain evolution, right?
Right.
And so, and those basic principles scale to size, right? So evolution can make things, can evolve things like the flagellar motor on one scale, but you can't propel a dinosaur with something like this, right?
Yeah.
You have to have a totally different way.
Well, in a way, they almost do with the mitochondria having a motor. It's, a whole different system, but many steps removed.
The mitochondria make the ATP that allows the muscles to work. That's true. But what it says is, which is I think really cool, is that mechanisms that you could not imagine working in a living organism of our scale, or even at a fruit fly scale. I mean, this would not work in a fruit fly, right? I mean, it'd be way too big. It can only work on something like a bacterium.
Yeah, I mean, not even most eukaryotes would be way too big.
Oh yeah. Eukaryotes that have flagella, like a sperm, for example. Their flagella doesn't rotate.
Even fungi that have
their flagella beats. It forms a helical wave, but it does it by, like you would do a jump rope, right? I mean, it does a helical beat. It isn't the rigid helix that gets rotated.
Yeah.
So the principles have to change. Design principles have to change. Also, if you're a bigger cell, you need a cytoskeleton. You need proteins that move along filaments, like microtubules and microfilaments and all kinds of things to get things transported distances in something as big as a eukaryotic cell. The fusion isn't quick enough, right?
Yeah.
So you need a different way of doing it. And that took a long time in evolution.
Yeah.
Bacteria were around for billions of years before you started getting bigger cells.
And so I guess one of the things I was building towards was like, do you think that those forces are strong enough to have these kind of motors keep independently evolving? Or do you think that, some billions and billions of year protist...
There is an example.
Yes.
All right? There are 3 domains of life, they say, right? There are eukaryotes, bacteria, and the archaea. Okay? Turns out the archaea also have helical flagellar filaments that rotate.
Okay.
The rotation is generated in a totally different way. The rotation is generated by ATP hydrolysis. Okay?
Wow. Okay.
So they have a totally different powering mechanism. Yet, the basic idea of rotating helical filament is conserved. Right?
So in a lot of, it's like, a lot of convergent evolution, but not homology.
That's a terrific example of convergent evolution, right?
And for
2 different lineages, 2 totally separate evolutionary lineages, which have been separate for billions of years, found a way to move by rotating the helical filament, right? And they could do that because they were small. That's what they shared in common. They were small, and it turned out evolution selected for rotating helical filament as a way. But the motor that powered it is totally different.
That's incredible. Yeah. I mean, because, when people hear about convergent evolution, like in high school biology or whatever, it's always these classical examples of like a bat wing versus a bird wing, right?
Yeah.
Similar structures, but not necessarily the same like actual development
Right.
into building those structures. But on the, protists and
But, I
micro
mean,
scale,
if
you
you
never
think about
that.
like birds and bats and pterosaurs.
Yeah. I mean, they're all tetrapods, which in a way.
Well, they're all tetrapods, and they all fly by moving their front limbs, right?
Yeah. I guess
But flight evolved in insects. It's totally different, right? It's totally different. They're not. They didn't turn a leg into a wing.
right.
They have different, totally different
appendages.
structures
Yeah.
that are wings, but they still have evolutionary constraints that make them funky. You have to
Right. They have
beat
to be
the
light,
wings.
they
You have
have
to
good surface
change.
area.
Yeah.
Yeah.
So
These like deep evolutionary questions are always really fascinating. to me.
Oh, absolutely.
And once you start getting to
And then you also have the question: okay, is it there are bound to be places in the universe that are conducive to the evolution of life. If life evolves there, how similar is it to what we have here? I mean, are there really relatively tight constraints that say, oh yeah, you got to have DNA, you got to, or are there other ways of transmitting information from generation to generation? I mean, it's like one thing you're probably going to have to assume is that you're not going to have things that live forever, right? So you're going to have some kind of reproduction. You're going to have structures that get old and fall apart. So you're going to have reproduction. But how you have two sexes? Do you?
I mean, even on Earth, there's many species that have way more. I mean, fungi have thousands of sexes.
Well, yeah. So, well, hopefully you can cobble together something out of these two interviews. And
thank you so much.
That's,
Thank you so much. We really enjoyed all these insights into this.
well, I really enjoyed having the opportunity to explain it.